Hello everyone, I'm Santosh Kumar and many of you are watching our Science and Technology series for preliminary examination on YouTube. This year we're running everything on YouTube. I have already completed space technology, nuclear science and technology, and now I want to jump to biotechnology. But before I do that, I want all of you to cover basics of biology. You may do it from NCERT, or I have requested Sonia ma'am that she will take some classes on biology basics, which will not only be relevant for your preliminary examination preparation, but would make it easy for me to cover biotechnology through when students have basic understanding, it becomes easier for me to cover biotechnology in a proper manner. So before I start biotech, please finish these classes. If you have done biology at your own, you may skip it, but I would recommend एक बार चाहे double speed पे just go through it, complete it, and then we'll start biotechnology classes. Thank you very much.
So yesterday we had started the cell part. Today we have to finish this complete handout. Before continuing the handout, we'll just revise quickly what we have done yesterday. We have started the cell part. We have said that on this earth, everything can be divided into living and non-living component. Living component is the interest of biology, where we study all the living parts. Now, for anything to be a living organism or entity, it should have a minimum basic structure, that is cell. So cell is the minimum requirement for anything to be called as living organism. Done? Now, cell therefore is called as fundamental unit of life, basic unit of life, structural and functional unit of life. Then we went on for structure of cell. We said that in the structure of cell, there is an outer membrane which is called as cell membrane or plasma membrane. Inside it, there is a control center which is called as nucleus, and there is a fluid space outside nucleus which is called as cytoplasm. Within cytoplasm, there are other cell organelles that perform the function of compartmentalization. Means there are several other compartments for cell to perform certain functions. Now, based on this nucleus, you have two types of cells. If the nucleus is primitive, then it is called as prokaryotic cell. If the nucleus has proper membrane and DNA is inside it, then it is called as eukaryotic. Prokaryotic cells are the cells of bacteria or archaebacteria. They are usually unicellular in nature, means they do not collaborate with other cells to form an individual. They'll collaborate with other cells, they'll form colonies, but that won't be called as single individual. For example, if our body is there, there are billions of cells who are collaborating with each other so that one individual can run. But in unicellular organism, in bacteria, they can live together. Several cells can live together, but they are not one single organism. Done? Next, coming to eukaryotic. Eukaryotic cells are found in fungi, protista, plants, animals. So most of the bacteria are, or usually all of the bacteria are prokaryotic and others are eukaryotic. So we as a humans have a true nucleus within ourselves and we are eukaryotic organisms. Next, within the cell, we have went for the outside plasma membrane. The outside plasma membrane, the function of this membrane is to protect it, to regulate what is coming inside, what is going outside. So entry and exit from the cell or within the cell is regulated with this plasma membrane. Whatsoever can come inside the cell without use of energy is called as passive transport. When energy is used, it is called as active transport. In passive transport, if transport is occurring of gases, of liquids without any membrane, then it is called as diffusion. If there is a membrane and water is moving from one end to another, then it is called as osmosis. So if concentration of water is outside more, it will come inside. It is called as, the cell will swell and the condition is hypotonic solution. If the concentration of water inside is high and outside is less, then water will move out of cell and the cell will shrink and the solution is called as hypertonic solution. If the movement is balanced from both the sides, it means it's same condition, same for same, we have a term called as iso and the solution is called as isotonic solution. Done up to here. Now next part is that for a cell, let's say this is a cell. In animals, you have a nucleus, you have other cell organelles lying, but in case of plants, plant cell has a cell membrane, they have a nucleus, but to protect it from extreme condition, they have one more outer membrane, one more outer layer, and this is called as cell wall, which is rigid. Means unlike plasma membrane was fluid, it was flexible. That's why we said for plasma membrane it is fluid mosaic model. But this membrane is not fluid, it is not flexible. That's why the term wall has been used, because wall is rigid and this wall is rigid. It is made up of, in case of plants, it is made up of cellulose. And cellulose is a type of carbohydrate. Simple. Okay. So in case of plants, we have an external cell wall which is made up of cellulose. Now what about other organisms? Do they have such cell wall? Yes. Even fungi. So if I take plants, we have cell wall that is made up of cellulose. If I take fungi, yes, they have cell wall which is made up of chitin. What about bacteria? Bacteria also have cell wall. Bacteria is a prokaryotic cell or eukaryotic cell? Prokaryotic cell. They have primitive nucleus, but they are again living in extreme conditions. They should have a wall around themselves to protect them. Therefore, bacteria also has cell wall, and this cell wall is made up of a compound called as peptidoglycan. You don't have to remember this, but based on this peptidoglycan, there is a function of antibiotics. You usually have studied antibiotic resistance also, right? So, whensoever, if there is someone who is hurting me, right? So, what I'll do is, whatever is the strength of that individual, I'll hit there. Now, if bacteria is causing disease in me, so I'll hit its strength. And what is protecting this bacteria? This cell wall. And if I hit at the cell wall, the bacteria will die. So most of the antibiotics hit this peptidoglycan. Most of the antibiotics hit this peptidoglycan layer. So bacteria has peptidoglycan cell wall, and antibiotics, usually your cephalosporins or penicillin or ampicillin, they break this peptidoglycan cell wall, due to which bacteria will die. Done? Understood? Now, there are some naughty criminals. There are certain bacteria who do not have this peptidoglycan cell wall in thickness, means thickness varies. In some bacteria, peptidoglycan layer will be, the wall will be very thick. And if you hit that wall will break and the bacteria will die. But in some bacteria, this wall is thin. So you cannot hit peptidoglycan because wall is thin. You might even understand wall is thin, so anyone can go and break. But the main layer where you have to work, that is only very thin, and additionally they have some other layer to protect themselves. So therefore, bacteria, what happens in bacteria is, there are some bacteria where the cell wall is made up of peptidoglycan and their antibiotics can work and kill them. Such bacteria are called as gram positive. You don't have to go in the detail why they are called gram positive, why they are called negative. That's just a stain work. If they take the stain positive, doesn't take the stain negative. That's it. Okay. My motto here is to tell you that why some antibiotics work on some bacteria and why they do not work on other bacteria. Some other bacteria, they have peptidoglycan layer, but separately they'll have one more layer, which again you don't have to cram, but given in your notes, due to which antibiotics cannot work on them, and such bacteria are usually gram negative. So most of the antibiotics which are available in the market work upon gram positive. They kill them, but they do not work on gram negative. It is the main reason is presence of peptidoglycan layer. That's it. Done. So function of cell wall, done. So we have done this plasma membrane part. We have done diffusion. Next is cell wall. So cell wall, in plants, they have a rigid outer layer which is called as cell wall. It lies outside plasma membrane made up of cellulose. Next, in plants, what we did yesterday was that when plants are kept in hypertonic solution, what happens? The cell membrane will shrink, but the cell wall will remain as it is. So due to which the structure of plant remains usually same. Here also, when they are kept in hypertonic solution, they will swell, but the cell wall will protect its structure and prevents bursting of cell. So plant cell will not burst. Otherwise, if you keep plant cell in the water, it will burst, it will come out, it will immediately wilt, right? It doesn't happen in case of plants such that because they have to survive very extreme conditions. Done. Now going back to cell wall. So this condition where plants are kept in hypertonic solution, the cell membrane shrinks, that is called as plasmolysis. Yesterday only I told that the word lysis means to break. So here plasma, the cytoplasm is getting shrinked. I can call it as plasmolysis. Biology is very easy if you crack the linguistics, means terms, meaning of terms. So if I say cytolysis, breakdown of cell. Phago is a word for eating food. Yesterday I said phagocytosis. Cell is eating something, right? So the terms are very easy here. Next is plant cell. They can withstand several changes. Why? Because of presence of cell wall. Next, bacteria has cell wall made up of peptidoglycan. Antibiotics like penicillin, they can work against gram-positive bacteria. Reason being because they have peptidoglycan, and most of the antibiotics work to break this peptidoglycan layer and they can kill the bacteria. That's it. The simple reason is this. Next is bacteria. They can have another structure outside them to protect themselves. So they have a cell membrane. Bacteria, they'll have outside layer. Apart from this, they can also have outside layer, one more layer, which is called as 𝐂𝐭𝐤𝐵, or it can also be called as capsule. Okay, so anything can be called. What does this layer help? Protection against foreign organisms. It can also help in adhesion, sticking to some other surface, because bacteria are on our body surface everywhere. They are omnipresent. So they can be sticking to other, they can interact with other bacteria. So they help in that. They also help in protecting or conserving the water. Dehydration se bhi bachate hai bacteria. Done. Next is fungi also has cell wall which is made up of chitin. Animals do not have cell wall. Favorite question of UPSC, animals do not have cell wall. Okay, done. So animals do not have cell wall, bacteria has, fungi has, plant. Exception is done. Next is nucleus. Nucleus is the control center of the cell. Yesterday we understood it with the analogy that all the important files, all the important information which is very private is kept in the nucleus. So what is the most personal thing to you is your information, your DNA, which is being transmitted to you throughout generations. Your parents has received their DNA from their parents and they are transmitting it. So what makes you unique? See, all the individuals on this earth, humans, almost 99% of their DNA is same. Okay, everyone has the same biological structure, same 2 hands, 2 legs, everything is same. What makes you you, that your characteristic physical features, your eyes, your hair, your color, anything, is because of your DNA. And if someone steals that DNA, your complete identity is being stolen, and that can happen in cloning as well, right? So that information is kept within the nucleus. So what is making you is kept within the nucleus. Nucleus has all the genetic information. Okay, what will be the color of hair? What will be the color of eye? How many hormones, or which hormone this person will secrete, which it will not secrete. Those all things are, even your height, growth hormone, everything is kept here. The information is here. And the language of that information, when I'm communicating with you, I have a language. I'm using English right now. So that information should be kept in some language. And the language of that information is DNA, chemicals. The language is chemicals, and that is DNA. And my body is made up of information is in the language of DNA, and my body is made up of proteins, carbohydrates. So this language has to be converted. If my cell has an information that this individual should have a brown hair, so that information should go somewhere and tell that this individual should have brown hair, and the brown hair should express. And the hair is made up of which protein? Yesterday you only told keratin protein. So that color should be expressed there. So this information is even translated. When I translate, for example, most of the individuals, and mostly many, or even all, whensoever someone is speaking in one language, most of us translate that into our mother tongue inside our head. Same way, that information is kept in DNA language, and for expression, it is translated into another language. We'll do it in detail, but after studying nucleus. Okay. Now, what is the structure of nucleus? The structure of nucleus is so if a cell is there, the structure of nucleus is that it will have double membrane, not single. I'm making single here, but here it is double membrane. So there are 2 layers, one this layer and one this outside there. So it is double membranous structure. Now from the chamber, you want things to come inside and out, right? So you will leave pores in the middle. It cannot be a rigid wall because information has to move. So there are pores. This is nuclear membrane. There are pores in the middle so that movement of molecules can occur. The genetic information, the DNA is lying here, but the information is scattered, open. For example, if I go to your room and I see the pile of books lying very properly, neatly arranged in a cupboard, what I'll infer from this? Will I infer that the person is studying, or will I infer the person is not studying? Not studying, right? And if the books are lying on the bed, right, open, pens are scattered, what I'll feel? This person is studying. So this information usually is lying scattered, open, in a thread-like structure. Okay, means the information is kept on reading. Someone is reading this information and using that information to run your body. Okay, done. So this genetic material is lying in a scattered form in a meshwork thread-like structure, which is called as chromatin. Which is called as chromatin. Done. This DNA, if I make it again here, so it's a information thread-like structure. So this DNA will have certain segments. Means, for example, your book. You have your book divided into several chapters. Same way here also. What happens is, there is lots of information lying here. Let's say this segment of the information is telling me the information that insulin has to be formed. Means the individual should form insulin. So how to form insulin, or what will be the structure of insulin, will be kept here. And if I take out this piece, I will get to know that how insulin will be formed. What will be the structure of insulin? This part of DNA, or this segment of DNA, which gives me an information of a particular thing, that is called as gene. So gene is not the complete DNA. It's a segment of DNA. It is not the complete DNA, but it is a segment of DNA which is giving me an information that how a particular protein will be formed. Done. So it's a functional unit. DNA is there, complete structure is there, but only few fragments are giving me some function. It is functional to me, it is useful to me. And that's why it is called as functional unit of information or inheritance. So if I carry a gene within me which is not working, which is not giving me correct information of how to make insulin. Okay, the information can be correct, it can be incorrect. So if I have received such a gene which gives me correct information, then my body will make insulin correctly. And if I do not receive from my parents this information correct, means it is incorrect, then will my body make correct gene, correct insulin? No. In that case, I'll have disease. So correct inheritance of genes is also necessary. Done. So within the nucleus, the information is lying in the language or in the form of DNA. The segment of DNA which gives me information to form a protein, that is functional unit, that is called as gene. And this complete information is lying in a scattered form which is called as chromatin. Now, your books are lying, your information, complete books are lying in the room. Now let's say you have to shift your room. Will you carry all the books open? No. You will pack them and then carry. So if my parents have to give me information regarding the genetic material, or if my cell has to divide, let's say my hair cell has to divide. So one more cell has to be formed. So this new cell should also carry the same information which this cell carried. So will it be carried in this scattered form? This has to be packed properly. And how it is packed properly is in this form. So the complete material will be packed into several structures. This structure called as chromosome. So can I say chromosome is compact form of chromatin? Because it is being packed. And what is being used to pack this is proteins. Okay, it is exactly like, have you seen those tailors use those thread, right? They are packed on the cardboard or a structure, right? Or a plastic cylindrical thing. So the thread is used, but packaging mein they are using that base over which the thread is wrapped. Same way here also our body uses proteins and wrap DNA around it to make these structures, which is called as chromosomes. Done? Understood? Any doubt up to here? Now, how many chromosomes? There are total 46 chromosomes. So, our body carries, or every cell carries 46 chromosomes. Is each chromosome unique? No. Chromosomes occur in pair. They occur in pairs. And how many pairs? 23. So there are 23 pairs. Just like you have shoes in pairs, you have chromosomes also in pairs. And how many pairs? 23 pairs. And these pairs are identical, just like your shoes are identical. ऐसे तो नहीं होता अलग अलग shoes पहन लिए, right? So similar way, the chromosomes are also identical. For example, if I have this chromosome, and on this chromosome here is the gene for, let's say, insulin making. So its pair will also have a complementary gene, means it will be similar, and it will also have, let's say this is chromosome number 1. We have not named the chromosome. We have 23 pairs. We say it as 1, 2, 3. Okay. So first, let's say first pair has a gene. So its complementary pair will also have same gene. What's the benefit of this? If one is not working, other will work. It's good if both are working, but if one is not working, other will work, right? And if both are not working, Bhagwan bharosa, right? Then disease will happen. Done. And if you have extra also, then also it's a problem. If you have one more of it, that's also a problem. Okay. So chromosomes occurs in pair. Humans have 23 pairs of chromosomes, out of which the last pair, that is for determining sex of the individual. If the chromosomes are X and X, that is for females, and X and Y, that is for males. Done. Nucleus. Any doubt up to here? Now going back, nucleus. So it's a control center of the cell. It stores DNA, the genetic information. Position of the nucleus is different. In case of animals, it is at the center and it is controlling everything. In case of plants, because plants do lots of work of production also, because they make their own food. Unlike us who are dependent on somebody else, plants make their own food and they are working in very other extreme conditions. So they have a storage box which occupies lots of space, that is vacuole. And because of presence of vacuole, their structure is different. For example, if this is a plant cell, because of presence of vacuole, their nucleus will be sidelined and it will not be in center, it will be on the sides. But in case of animals, it will be in the center, usually. Okay, done. So plants they have a central vacuole, because of the presence of which they pushes a nucleus to sides. In animals, a nucleus is at the central position. Now, it is not necessary that individual will have or cell will have one nucleus, multiple nucleus. It may also be their choice that cell do not have any nucleus. It doesn't want any control center, because now it is very specialized. It will perform only one work. That's it. Once the work is done, its life will be over. It doesn't need to give information to somebody else. It is वो होते हैं ना, suicidal bags. Suicidal bags is the wrong term here, because suicidal bomb बस एक काम किया और खत्म. Same here also, that a cell might have nucleus, it might have multiple nucleus, it might even not have nucleus. It is not necessary cell will have nucleus always. So some cells of your body will have one nucleus, some will have multiple, and some may not. If nucleus is one, for one we have a term uni, so it will be called as uninucleate cell. If the cell has multiple nucleus, multinucleate, and if it does not have any nucleus, anucleate. When multiple nucleus will be there? When cell has to do lots of work. या बहुत सारी पढ़ाई कर, बहुत सारे दिमाग होने थे, या बहुत सारा टाइम होना था. So when you need multiple control centers, because you are doing very important work and you need the information multiple times. For example, I had Lakshmikant too. I had two Lakshmikants with me, the older version and the newer version, because I read it multiple times. So my previous book was torn. Same way, nucleus can be multiple, because if the cell needs information very fast, so more nucleus are there, so that multiple times it can be read and the information can be processed. So same here, multinucleate cell will have more nucleus, usually occurring in muscle cells, because your muscle cells are continuously working. Apart from that, even liver cells have two nucleus, because they are continuously working. They are digesting your food. Whatsoever medicines you take, it is digesting it. Detoxification is done by liver, so it will have multiple nucleus. Cells like RBCs, they do not have nucleus. Their nucleus degenerate, means they remove their nucleus. They don't want it. Why they don't want it? Because they are so specialized that I'll do this work only of transporting oxygen. RBCs transport oxygen, right? Red blood cells. Why? Because they have, RBCs have hemoglobin. And the function of hemoglobin is to transport oxygen. So RBCs say, I'll do only one work, that is transporting oxygen with the help of hemoglobin. So I don't need nucleus. So they remove or they degenerate their nucleus. Now, are they carrying the information of the cell? No, they are not carrying, because they do not have nucleus. So they are not carrying the genetic information. So can these cells multiply? No. No, these cells, what I said, they are just for that function. Once they can do, they will be doing, then they will die. They will not reproduce. Why? Because they are not carrying the information. What they will give to their offspring cells? Because they do not have any information. They chose to remain single. Right? So they are not carrying the information. In that case, these cells do not divide. So mature RBCs, nucleus should be somewhere here. So if I scoop this part out, the cell shape, it will be hollow from inside. So the cell shape is biconcave. Why I'm telling you this is because in biotechnology you will be reading about sickle cell anemia. So usually RBCs have biconcave shape. Why biconcave? Because it will be hollow from inside, because nucleus is removed. So from both the side, if I see it from sides, how it will be appearing with me is something like this, because from the center nucleus is not there. But sickle cell anemia, you'll have the RBC shape in the form of sickle. Done. So RBCs, what they do, they perform their function of transporting oxygen for some time and then they die. They do not multiply. Done. Any doubt up to here? Done. Next is another example here is in case of plants, sieve tube elements, they also remove nucleus from themselves because they want to make a channel. We'll do tissues today. We'll see sieve tubes. So since tube is there, so it should be hollow from inside. So they remove their nucleus so that it can remain hollow and perform the function of transportation. A nucleate cell cannot divide because they do not have any information and thus have shorter lifespan. They'll die after performing their function. Next is in case of prokaryotic cell, the nuclear membrane is not there. Nucleus is primitive. That's why they are called as prokaryotic. And the genetic material is poorly lying in the cytoplasm and thus called as nucleoid. Structure of nucleus, it will have a double layer covering. It will have pores for transportation so that material can move in and out of it. Apart from that, their outer membrane is continuous with endoplasmic reticulum and they will have again a fluid-like structure within it that is called as nucleoplasm. Just like for cell, we had cytoplasm. For nucleus, we'll have nucleoplasm. And there is also a controlling structure within the nucleus also, which is called as nucleolus. The information is lying in the form of chromosomes. What are these chromosomes? They are made up of DNA and protein. Just now I told the information is in the form of threads called as DNA. This DNA is wrapped over protein. So chromosome will be formed for both the things, DNA and protein. So if the statement comes that chromosomes are formed only of DNA, it's wrong because for packaging, proteins are used. It contains information for inheritance. DNA is there for carrying the information of inheritance. Functional segment are called as gene because it gives me that one particular protein structure. Humans have 46 chromosomes. 46 pairs are not there, 23 pairs, 46 chromosome. What is the structure of chromosome is? It is like this. The center part, this center part is called as centromere. These are two arms of it and these are end part of it, telomeres. Telo word is for end. Ethics में पढ़ा होगा teleological for end gains. So ends are called as telo and the part is called as telomeres. Done. Next is chromatin material. So when the cell is not dividing, it is resting, means it does not want to carry the information to the next part. In that case, the information is lying scattered in the form of chromatin, and if it has to do transfer, then it will be packing it in the form of chromosomes. Next is chromatin material is entangled mass of thread-like structures. So whensoever they divide, they organize into chromosomes. Another thing which is there in nucleus is nucleolus. So cell has nucleus. Within nucleus there is nucleolus. It is made up of RNA and protein. So there are several RNAs and protein. What is their function is that ribosomes. Yesterday we studied a body which helps in making proteins. So ribosomes are formed from, a part of ribosomes are formed from here. Okay, they help in making ribosomes. So function is of the nucleus is that storage and protection of genetic material. These three functions will do later when we'll completely compile all the information that how cell is working. Right now we are doing just structures, what is present in cell, then we'll do functioning of cell. Next is cytoplasm, which you have already done. It's a fluid content inside the plasma membrane. It contains water, enzymes, nutrients, salts, etc. And in eukaryotic cell, it also has specialized organelles which perform their individual function. These organelles have a membrane outside them. Why? Because of compartmentalization. You want everything to happen in a particular area. So membranes are there in eukaryotic cell, helps in compartmentalization. Membrane forms the boundaries. Next is viruses. They lack these membranes. They do not have proper enzymes to be formed. That's why they are non-living. And to multiply, they have to have the structure and they go inside the host cell to remain living. So viruses are the like that bhatakti atma. सब आर पार निकल जाता है, कुछ नहीं है उसमें. It has to go and rest in some other body to remain alive. Next is what is there in the cell. So we have done yesterday the part that cell, apart from having a plasma membrane, a nucleus, other specialized structures, within it, it has a membranous structure which is continuous with the nuclear membrane. I made nuclear membrane, and with this other membranes are continuing. This membranes along with other membranes. There are other membranes. What was this? Endoplasmic reticulum, because it's a network, net-like structure. In biology, the term is reticula or reticulum. It is within the plasma, so endoplasmic reticulum. Apart from that, for packaging, we had Golgi bodies. It also has boundaries, membranes. Apart from that, we had lysosome, which contains certain chemicals which helps in breakdown. Another membrane is for vacuole. Vacuole is for storage. So these are membranes, membrane, boundary, membrane, boundary, membrane, membrane. So all these are membranes. Can I call them as endomembranes? They're within the cell, and when we'll do functioning, they all are working together. Information, production, packaging, storage, and this is for whatever is coming from outside to open it up, to break it down. So they are working in tandem, means in synchronized way, organized way, in a system. So can I call it as endomembrane system? So endomembrane system is composed of endoplasmic reticulum, Golgi bodies, lysosome, and vacuoles. So now we'll move on to this endomembrane system. Done? समझ आ रहा है? ज़्यादा हो गया फिर से? ये तो कल कर लिया था. We are just reading it. फटाफट से. So endomembrane system, the group of membranes in the eukaryotic cell, helps in modification, packaging, production, etc. First part is endoplasmic reticulum. Their main function is synthesis. Synthesis means production. Production of what? Proteins and lipids. So if they have these dot-like structures over them, which are called as ribosomes. Ribosome function is to make proteins. So they will make proteins. They are appearing rough. So rough endoplasmic reticulum helps in making proteins. Why they help in making protein? Because they are associated with ribosomes attached to their surface. So production of proteins. The smooth endoplasmic reticulum, which do not have ribosome, they help in production of lipids. Easy way to remember is smooth. So lipids are also smooth, greasy. So smooth endoplasmic reticulum. Next is it has large network of membranes which are bound by tubes. So there are tubes in the middle, and these network of membranes are present which are giving the production site area, and sheets are also there. This looks like sheet-like structure, tubes and sheet-like structure. Oblong vesicles are there. They are continuous. The membrane is similar to the structure of plasma membrane. What does this mean? Yesterday we did that plasma membrane is made up of lipids and protein, similar. Bilayer, similar structure of endoplasmic reticulum. All the membranes are same in nature. Done. They are absent in prokaryotes. Most of the cell organelles are absent in prokaryotes. RBCs also do not have them. Why? They are specialized, they have hemoglobin and they are chilling in transporting oxygen. Cells that synthesize and release lot of protein will have more ER obviously, because they need more ER, because production has to be done more. Two types of ER, rough endoplasmic reticulum. Why they appear rough? Because of presence of ribosomes on their surface. Their main function is to manufacture proteins. It looks like sheets or disc of bumpy membranes. Once they manufacture proteins, they will send it to Golgi bodies for packaging and modification. They are attached to nuclear envelope or nuclear membrane. So I made it in continuity with the nuclear membrane. Smooth endoplasmic reticulum, manufacturing of fat molecules or lipids. Their shape is again the same. So together, both of them help in lipid and protein production. This lipid and protein is needed for membrane formation also. So for production, in biology, we have a technical term, genesis. And what kind of genesis? Biogenesis. Whose biogenesis? Membrane biogenesis. So can I say ER is helping in membrane biogenesis by making proteins and lipids? Because these protein and lipid will be used to form the surface. And other proteins are also formed. It's not like only for membrane. It will make proteins of all other kind. Okay. Next is, although ER varies greatly in appearance, it always forms a network of structure. Apart from production, it also helps in transporting, because iska jaal pura dur tak bichha hai. So it will also helps in transportation of material. Next, ER also functions as cytoplasmic framework, because network is spreaded throughout the cell. So it will maintain the cell structure also, because it is present inside it. The ER also surface for some of the biochemical activities. Yes, we have done protein, lipids, iska production karwa raha hai. In liver cells, smooth endoplasmic reticulum is important, because liver is doing the function of detoxification. And for detoxification, you need certain chemicals, and those chemicals will be produced by ER. Next is Golgi bodies. Golgi bodies, why they are called Golgi? Because the discoverer was Golgi, on whose name the name is given. Again, it's a meshwork-like structure. So what happens here is, if the cell is there, let's say nucleus is this side, this is endoplasmic reticulum. So Golgi body will be lying next to endoplasmic reticulum. Let's say ribosomes have produced a protein. A protein is produced by ribosome. This protein will travel all through here, will enter Golgi body from here. Okay, so it will enter. So they have mentioned incoming transport vesicle. So it will come from here. The protein will be modified if certain modification is required. Then it will be packaged, and then it will be leaving from this side. That's why secretory vesicle or newly forming vesicle. Done. You don't have to cram cis-phase, transphase. It's not needed. You just need to know that packaging and modification is done in Golgi, and why, how. Information is read by ribosomes. Ribosomes make protein from ER. The protein will move. It will go to Golgi body. Done. Next, they are parallel to ER. They are made up of certain cisternae and tubes, vesicles. Function is storage, modification and packaging. For some time it will store the protein which is coming from ER, it will do modification, it will package. How modification? Some sugars it will add, right? Next, it also involved in formation of lysosome. How? Because these proteins, some of these proteins are also helping in breakdown. So lysosome carries those proteins and inside that it will help in formation of lysosome. फिर से ज्यादा हो गया क्या? पीछे वालों के तो चेहरे ही नहीं दिखते यहाँ तक. After making these big molecules, Golgi apparatus packages them into vesicles. Next is lysosome. See, if a cell is carrying certain chemicals which is breaking down certain products like carbohydrates, proteins, fats. So if I am carrying certain chemical which has an ability to kill my own structures, because my body is also made up of carbohydrates, fats, proteins, and I'm carrying that structure also which can destroy all these three. So will I keep it scattered or will I keep it in some proper vesicle thing? If you are keeping acid or phenyl in your house, so you're keeping it in a bottle, right? Proper bottle, packaged thing. You won't keep it lying. So same happens in the cell. So cell keeps these chemicals. These chemicals are called as enzymes. First of all, these chemicals are called as enzymes. Enzymes has the ability to conduct or to create a reaction. Enzymes facilitate a chemical reaction. So any chemical reaction which is occurring in your body is occurring because some enzyme is there which is helping it to occur. Now some of these enzymes can also do destruction, right? So these enzymes should be lying in the cell or should be kept in a separate compartment. In a separate compartment. So that compartment is lysosome. Why lyso word? Because these enzymes has the ability to break. For break we had the word lysis. So this body is carrying certain chemicals which has the ability to break lyso, lysis and zo. So it is called as lysosome. Done. So lysosomes are membrane bound because it will have membrane. You have to keep it somewhere. Sacs which are filled with digestive enzymes which are capable of breaking organic material. These membrane ensure that these enzymes do not kill the cell itself. And if they come out, if this membrane within which lysosome is there, if this membrane breaks and these enzymes comes out, what happens? Cell will die. And due to this reason, these are called as suicidal bags. Due to this reason, they are called as suicidal bag. So if a cell wants to die or if cell wants to commit suicide, it will break the lysosomal membrane, all the enzymes will come out and they will scatter and break down the cell. So lysosomes are also called as suicidal bag. Originally, what should be their function? That whatsoever is coming from outside, the food should go here, the chemical should react, the enzyme should react, and the food should be digested. That's it. Next, what are the major enzymes? See, your body is made up of protein, carbohydrate, fats, and nucleic acids. It will break all these. Usko jaga do bhai. हाँ. So your body is made up of proteins. So for proteins, there is an enzyme called as proteases. Proteins are made up of amino acid. So everything, every compound is made up of some small unit. So बहुत सारे amino acid, lots of amino acid will together make protein. So if I have to break down protein, again it will make amino acid because it is made up of amino acid. So proteases will break down protein into amino acid. Lipases will break down lipids into fats. Nucleases will break nucleic acid, that is DNA and RNA, and glycosidases will break carbohydrate. Simple. कार्बोहाइड्रेट ग्लूकोज से है तो वैसा ही मिलता जुलता वर्ड होगा ग्लाइकोसाइडेजेस. न्यूक्लिक एसिड के लिए मिलता जुलता वर्ड न्यूक्लियेजेस. प्रोटीन्स के लिए प्रोटीएजेस. लिपिड्स के लिए लाइपेजेस. डोंट हैव टू क्रैम. डन. फंक्शन क्या है? वेस्ट डिस्पोजल. अगर बाहर से कुछ भी आएगा, इफ अ सेल इज ईटिंग, लेट्स से बैक्टीरिया has entered into the cell. Bacteria is also made up of these four things: carbohydrate, protein, lipids and fats. It will come inside the cell. It will, lysosome will work, break it down, चिथड़े उड़ा देगा बैक्टीरिया के, and whatsoever are the debris will be, the dead body will be removed outside. Done. So this is how lysosome work. So lysosome is like a waste disposal system. It helps in digesting foreign material. They are called as suicidal bags because if the material comes outside, it will be. Next, they are not commonly found in plant cell because plant cell have a tough cell wall which protects it. Animals do not have cell wall, so anything can enter in our cell, for which we need certain compounds for its destruction. Next is vacuole. Vacuole is a large space which is kept for storage. So whatsoever functions or storage we have to do, we'll keep it in vacuole. We do not produce much, so we do not have much storage. But plants do lots of production because they are autotrophic organisms. They make their own food. So they need lots of storage and their vacuole is large. Logical enough? Function is storage sacs for solid and liquid content. Storage can be of amino acid, sugars, organic acid, protein. So whatsoever is there, that is being stored. Because vacuole has consumes a large space of plant cell, it also helps in maintaining its shape and structure. Because if vacuole concise, the plant will also shrink. Plant cell will also shrink. Next is ribosomes. Ribosomes are the one which are present on RER, and they can be also present scattered in the cytoplasm. Their main function is to make proteins. Now, what is the structure of ribosome? Are ribosomes counted in cell organelle? No, because they do not have membranes. So, what are ribosomes made up of then? So, ribosomes do not have membranes. Ribosomes are made up of two things, RNA and protein. Simply, you can remember R se ribosome, R se RNA. So, it is made up of RNA and protein. So, two things which makes ribosome is RNA and protein, which there are several types of RNA. So, the ribosome is made up of specific type of RNA called as ribosomal RNA. Simple. So, several types of RNAs are there. The RNA which will make ribosome is called as ribosomal RNA. So, I can simply in short form call it as rRNA. So, ribosome is formed of rRNA and proteins. Done. Their function is to make proteins. Now we'll study how. अभी तक तो यही कर रहे थे. न्यूक्लियस के अंदर इंफॉर्मेशन है. Something outside is there which is reading this information and making our proteins. Right? So, what kind of information is present in the nucleus will decide what kind of proteins are formed and how you will have appearance. So, if somebody has brown hair, the information is there in the nucleus. Ribosome has read it the same way and made the protein in the same way. So, the production site is ribosome. Now, going to how it is done. Simply a 2-minute process, easy. See, if a information is very personal to you, again and again I'm using this word. Why? Because there is something called as DNA fingerprinting and other stuff, and nowadays gene cloning has made very easy to replicate somebody, right? To make clone of somebody, and that's why we have personal data protection and everything. And DNA should be the part of that. Just like your fingerprints were your personal data, now your DNA is also personal data which can be misused. So again and again I'm using this word, that private information, personal information, Because nature in its system has recognized this. Cells do recognize that this DNA is very important and it cannot be shared. It cannot be shared. So its language is very specific and it is not shared with something else. Means this DNA will not come out of nucleus. जैसे हम बोल रहे हैं ना डेटा प्रोटेक्शन में that information should not go outside India. हर कंट्री यही सोच रहा है. ऐसे ही नेचर ने ये सिस्टम किया है that this information, this Pusa information should not go outside nucleus. But the production site is in cytoplasm. Information is in nucleus. This DNA cannot go outside. Ribosome cannot come inside for production. So this information has to be shared, right? How ribosome will get to know what kind of protein to form? Because information is lying inside nucleus. Do you need some mediator here? That something should be there which will carry this information to cytoplasm so that it can safely reach there, right? So if I have my complete information with me, there is important fact here that all the cells of your body which has nucleus has same genetic material. So if I have pancreatic cell, it will have complete DNA of mine. If I have my hair follicle cell, it will have complete DNA of mine. Hair follicle is not making insulin, but it will have that gene there. So all the DNA remains same across all the cells of your body. So if I steal your hair follicle also, I'll get all the information of yours. All the information. So if my hair follicle cell have to make keratin, so do I need to give ribosome complete information of mine? No. Its function is to make keratin. So take information of keratin. I won't give complete information to it. Right? Same way cell also does. See how beautifully nature works is that पूरी protection भी कर लेंगे और information उतनी ही पहुंचाएंगे. The information will be shared that much only which is required. Not anything extra. Done. So how it is done is cell, nucleus. Within nucleus, DNA is lying. Let's say I'll just give basics of biotechnology here. I'll connect so that sir can continue. Okay. This information is lying here. Now let's say this is a pancreatic cell. Pancreatic cell function is to make insulin. Now this cell needs to make insulin and this fragment here carries information of insulin. I don't want my complete information to go out because this cell needs only information for insulin. So let's say this is insulin gene. And genes are made up of DNA. And I said DNA is a language, means it will have certain alphabets. Just like English has 26 alphabets, DNA will also have. And the alphabets here are ATGC. ATGC means DNA is almost same, but the alphabets, the language, the chemicals are different. There are 4 ATGC. Means complete information within this DNA is written in these 4 alphabets only. For example, your book, your handout is right now written only in 26 alphabets. Right? I've used only those 26 alphabets to communicate in different combinations. Same way here, body cell uses different combination. It may use AAA, it may use AT, 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 it may use ATG, ATG, ATG. It may use AT, 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 GC, GC, GC. So different combinations it will use from these 4 alphabets. Information is here. It has this information of the arrangement of alphabet. But now this information should not go as it is outside. Why? Somebody can misuse it. I want to keep it private. So I'll keep certain code. For example, you people, when you want to talk about someone else and you don't want that person to know, so you keep some names for that. The other person doesn't know. अगर मुझे किसी की चुगली करनी है, कुछ बातें करनी है उसके बारे में और उसे पता ना चले, I'll keep some nickname for it. उसने ऐसा कर दिया या कोई कोड रख लेंगे. Same way cells also do this. Cells also do this. Okay, what they do, एक अल्फाबेट चेंज कर देते हैं. इंफॉर्मेशन विल रिमेन सेम, टी की जगह यू यूज कर लेंगे. तो जहां जहां टी है वहां वहां यू. मीन्स इन प्लेस ऑफ टी दे विल स्टार्ट यूजिंग यू. सो इंसुलिन का जीन क्या करेगा? व्हाट इट विल डू, दिस जीन इफ इट इज कैरिंग इंफॉर्मेशन ए टी सी जी सी, so instead of T, what they will make a copy of it. They'll make a copy of it, and instead of T, they will write. If it is stolen also, somebody else don't know what is the code. We humans have detected it, but cell at that time didn't know that humans will be so smart and they'll detect it. But they have used this code. They've replaced T with U. They'll make copy of this gene. Instead of T, they will put U. Now the language has changed. Instead of DNA, it is called RNA. Now the language has changed. Instead of DNA, it is called RNA. It is just like English and French, right? या फिर English and American English, S और Z का जो फर्क होता है, S की जगह Z लग रहा है वहां पे. Same way here, ATGC की जगह they will put AUGC. This is called as RNA. Because RNA has U, DNA has T. Only this difference is there. This will be copied. Now this fragment will come outside with the information AUGC, let's say, or AUCGC. Ribosome will read this information. How? Ribosome is like this. It has two units, one and the another. This information will be kept here. It will keep on reading. It needs another RNA which helps it. Now I'm doing translation. I'm translating from one language to another language. So the language is of nucleic acids, and I want to make protein. Why? Because ribosome function is to make protein. Now protein ki languages, alphabets are, protein are made up of, just now we did, amino acids. Proteins are made up of amino acid. There are 20 amino acids. So in the language of protein, there are 20 alphabets. In the language of nucleic acid, there are 4 alphabets. So this language has to be converted into the language of protein. Process is simply called as translation. If you translate from Hindi to English, you call it as translation. So Hindi has lots of alphabets, English has 26 alphabets, use different combination. Same way cell is using different combinations. For example, if Hindi has lots of alphabet, it is A and A. English doesn't have. Different combination. I use it multiple times. No, every cell in your body will have same information. Okay, so if I take hair follicle, pancreatic cell, skin cell, any cell which has a nucleus will carry same information. Why? Because only that part of information is being shared with ribosome. So तेरा काम ये है, तो ये information मिलेगी, इससे ज़्यादा नहीं मिलेगी. अगर pancreas, if pancreas function is to make insulin, it will get information of only insulin. Why you want other information? It's my private information, I'll not share. Right? If thyroid cells has to make thyroxine hormone, it will get information of that hormone only, nothing else. If my skin has to make keratin protein, it will get information, ribosome will get information of keratin only. Right? Understood this part? So that's why cells have divided. Cells have divided, it's like group study. You all want to clear UPSC, you all have same books. We will not read all the books, polity, geography, economy, then share. Right? It's like group study that cells have divided their job. They have all information together, but I'll read pancreas ka information, I'll read hair follicle ka information, some other cell will read some other information, and they'll collaboratively work so that individual can function. Understood this part? What we humans have did, we have decoded this nature's language and we manipulate this information. What is biotechnology is, if a bacteria is there, I know, and the interesting part is all the individuals are running in same language. All the individuals, whether it is bacteria, whether it is prokaryotes, eukaryotes, anyone, they're running on same language. And we humans are so smart that we can dupe even nature. So we said, okay, bacteria, you also have DNA. And your DNA is also carrying the same language, ATGC. Your ribosome will also read RNA language of AUGC. What if I remove your information here and I put some other information which I want you to read? Let's say my body is not producing insulin. Okay, I cannot produce insulin and that's why I'm diabetic. Somebody else has to give me insulin. If I take it from humans, it's ethically wrong. If I take it from some other animals, earlier they used to take it from pigs, but that creates a inflammation or allergic reactions. So what I said, bacteria, I'll use your machinery, I'll add my insulin information to your insulin, your genetic material. Now your cell will read this information and will produce insulin. I'll use this insulin and sell it in the market and use it for therapeutic purpose. So we have decoded this thing, that how language of carrying information is same, how the process of translation remains same, and how we can want other individuals to remain to function for us. कोई PSIR का है यहाँ? If somebody is of PSIR, they would know Gramsci, how information is being changed, how we are manipulated every time so that we can work according to someone else. We humans are also doing this for other organisms. We are manipulating their information. If you cannot work for us, we'll give you information, read that information, do our work. So that is complete basis of biotechnology. Biotechnology rests on this principle only. Understood this part? Done. So when a copy is formed from DNA to RNA, it is copy के लिए एक और technical word use करते हैं, colleges में भी use करते हैं, transcript, right? So when copy is being formed from DNA to RNA, the process is called as transcription, and when it is translated or proteins are formed, the process is called as translation. And with this, expression will be there. So whatsoever this cell expresses depends upon this information which rests in DNA, which is transcripted to form RNA and translated to form proteins which it expresses. So if I have brown hair, it is because in my genetic material, the keratin protein color has been said as brown. It carries information as brown. It will be transcripted to form RNA to make brown keratin of that particular color and translated by ribosome to form brown keratin, and that's why the color is that. Understood this part? Paka? Complete biotechnology rests on this. If we humans couldn't decode it, We couldn't do anything. Interesting part is an Indian has done this decoding. 𝐂𝐿 Singh Khurana. He got to know how information from RNA is being translated to protein. So if I want to make my own protein of my interest, I can go reverse also, because now I know translation. If from English A can be said as A, I can do reverse also, right? So 𝐂𝐿 Singh Khurana told us that this is the mechanism which cells are using, and that made biotechnology completely possible. Done? So this is the complete process, that there is this information which is lying in nucleus. It is converted into RNA. Now I said RNA are of various types. The one which is carrying message, I can call it as messenger. So it's a messenger RNA, mRNA. The one which will help in translation will be called as translator RNA or tRNA, which is giving site, that is ribosome. Ribosome is also made up of RNA. कौन सा RNA? rRNA. Simple. तो सबका पहला अल्फाबेट उठाया और RNA की टाइप बना दी. tRNA, mRNA, rRNA. Why only RNA? इतना RNA क्यों कर रहे हैं? Because RNA is very unstable. You want some person, give the information, die. Don't stay there for long time. Okay. So RNA is very unstable. It is very prone to reactions. It is very prone to chemicals. It is very prone to temperature even. So what it will do? Just quickly go to ribosome, give information. If it would have been stable, I don't want it, because I don't want my information to remain there for a long time. It is exactly like cryptography, right? कि बस information लिखी, दी और मिट जा. वो कौन से धुरंधर में शायद दिखाया है ऐसा कुछ, किसी मूवी में दिखाया है, लिखा और मिट गया. Same. See how nature is working. Nature is very smart. हम लोग अभी डिटेक्ट करें, नेचर ने सदियों से कर रखा है ये चीज. एंड इंटरेस्टिंगली एवरी सेल इज डूइंग दिस. Every cell is doing this, and we humans are manipulating it from past 50 years. Okay, done. So this is complete basis of genetics, cell expression, gene expression. Now is powerhouse. How cell is doing all these functions, so many work it is doing, from where it is getting energy. So for energy there is a powerhouse called as mitochondria. Mitochondria is called as powerhouse of cell. It is burning the food. How it is burning the food? Yesterday I told digestion and respiration. So we need food, we need oxygen, right? So what mitochondria does is it breaks down the food completely in the presence of oxygen to release carbon dioxide. बचपन में पढ़ा होगा respiration, photosynthesis भी पढ़ा होगा. So even nature is again very smart. मतलब हद होती है नेचर की स्मार्टनेस की. Plants, they are taking oxygen, sorry, they are taking carbon dioxide using sunlight. मतलब सारे के सारे, वो कौन सी मूवी है ऋतिक रोशन की जिसमें वो धूप से एनर्जी लेता है? जादू. सारे प्लांट जादू बने बैठे हैं. Right? They are taking sunlight energy, taking carbon dioxide, making it into glucose. We animals are so selfish. We are taking that glucose, using oxygen, burning it, again releasing carbon dioxide. Do you think it's a basis of complete environment? And when you read global warming, you are disrupting this part, right? You are taking lots of oxygen, but nobody is there to take carbon dioxide, and you say ecological balance is disturbed. Again, this forms the basis of environment, global warming, right? And that means the word ecological balance, that plants and animals should be in balance so that plants can take carbon dioxide, make glucose, we animals can have it, take oxygen, burn this and release carbon dioxide. And this is what is mitochondria doing. Mitochondria is taking food, it is taking oxygen, In the presence of oxygen, it is burning that food to release lots of energy. Now this energy has to be stored. It is usually heat energy, but it has to be stored in some chemical form. And that chemical is ATP, adenosine triphosphate. Don't have to cram it again. ATP. Storage is in the form of ATP, and you can use these ATPs to perform your function. It is just like you did some work, but let's say you did some work for someone and you don't want other person to pay you right now, or a barter system, you don't want other thing right now. So what you will say, give me something to store so that I can use it anywhere else. That is money. What money is doing, currency is doing. Same way ATP is just like currency. I have produced heat energy, but I do not want this heat energy to use right now. I want it to store for future purpose. So I'll store it in the form of ATP, and when I'll need it, I'll use this currency. This is what mitochondria is doing. Mitochondria structure is like, it has double membrane. One outside membrane is there, one inside membrane is there. So this is outside membrane. Inside membrane is very curved or has lots of infolds. Why? Increase surface area. जितना surface area होगा उतना reaction ज़्यादा होगा. So mitochondria is enfolded, and they again interesting thing is they have their own DNA and ribosome. Interesting part is why they have their own DNA and ribosome. Interestingly, it is said कि ये कभी time पे in many years back during the process of evolution they were also bacteria. We humans means animal cells thought, let's take this bacteria inside us, give it shelter, and it will produce energy. Okay, so we had the cells, the eukaryotic cell has taken it inside. Now it has remained a part of our cell only. We call it as mitochondria. It produces energy for us. Done. So they have their own DNA and ribosome. Logic is that why they have, because they were once a bacteria. Next is functions of mitochondria. They are called as powerhouse of cell. They store energy in the form of ATPs after combustion of food. Energy is formed during aerobic respiration, means in the presence of oxygen, and there is a lot of energy which is produced in the presence of oxygen. When oxygen is not there, the energy amount is less. Okay, so cellular respiration means presence of oxygen. Next is ATPs are known as energy currency. Cell need more energy, and therefore the cells which need more energy has more mitochondria. Inheritance of mitochondria. See, mitochondria have their own DNA, right? Now what happens when offspring is made? During fertilization process, there is egg and sperm. Now sperm is a single cell. Why its shape is like this? Because the cell organelles has rearranged itself. What does this mean? Nucleus has come here. Mitochondria has arranged somewhere here at the neck. Why? Because it wants to produce energy and propel it to move forward. And egg has a cell, has a nucleus where information is there. And other cell organelles like mitochondria, Golgi, everything it has of its own. What is the contribution of sperm is just to this nucleus. It gives only this nucleus, nothing else. Nothing else. Done. So now when sperm is giving nucleus of its own, so this cell after fusion will form a single cell called as zygote, which has nucleus fused together, one from mother, one from father, but other organelles are derived from Mother. So mitochondria is also derived from mother. So if mother's mitochondrial DNA has certain issue, will the child have? Whether daughter, whether son, irrespective of gender of the child, if the mother has certain genetic issue in mitochondria, then the child will also have. And such diseases are called as mitochondrial diseases, and they are inherited from mother to offspring. How to resolve this issue? My cell has lots of mitochondria. How I'll replace all the mitochondria? एक एक को जाके पकड़ो, एनकाउंटर करो सबका, नए डालो. इजी पार्ट इज न्यूक्लियस एक ही है, इसी को निकाल के किसी और में डाल दो. Done. It is called as mitochondrial replacement therapy, but interestingly, mitochondria are not replaced here either. Before fertilization, mother ke, if I know this mother has a mitochondrial disease, so what I'll do is I'll put her nucleus, I'll take out her nucleus, because nucleus is one, it's easy to catch, its size is also larger. So I'll take out its nucleus, put it in some other donor egg, and fertilization will put here. So I can do it before fertilization or after fertilization. I'll take out this nucleus, because this nucleus is now fusion of mother and father. This is called as pronuclear, and I can transfer this nucleus into some other cell and it will form offspring, zygote, embryo, right? So I can do it before fertilization or after fertilization. In 21, UPSC has asked this question, right? So if a mitochondrial disorder is there, it can be recovered, it can be corrected. There is a therapy for it called as mitochondrial replacement therapy. Interestingly, mitochondria are not replaced in this. Nucleus is exchanged either before fertilization or after fertilization. Done. Easy. Any doubt? दो केसेस में डाउट नहीं हो सकता, या तो सब समझ आ रहा है या सब आ रहा है. आगे वालों का तो समझ आ रहा है, पीछे वालों का मुझे नहीं समझ आ रहा है. आगे वालों का मुझे समझ आ रहा है, पीछे वालों का मुझे समझ नहीं आ रहा है कि तुम्हें समझ आ रहा है कि नहीं आ रहा है. So to prevent this inheritance, we can do mitochondrial replacement therapy either before fertilization or after. When it is done before, it is called as maternal spindle transfer, and when it is done after, it is called as pronuclear transfer. Interestingly, consecutively for 2 years, UPSC has asked this question. Consecutively for 2 years. There can be any type of disease. For example, during my research, I was working on one mitochondrial disorder which is related to eye, ophthalmoplegia, which was called, or it can be any thing related to mitochondria, because mitochondria itself was a bacteria once. So when even bacteria will have lots of genes of its own. So it was capable of independent existence once. Now it is not, right? Next is plastids. So plastids are something which are found in plants. So it is not found in animals. Plastids are just like mitochondria. In plants, plastids are there. They have different functions, different colors. If they have colors, they are called as, they will have a pigment or a body called as chromoplast. If they are green in color, chloroplast, which performs the function of photosynthesis. Why green color? Because green color, if there is a presence of green color, it is absorbing all other light excluding green. That's why it is appearing green. So it takes energy from all the sources of light except green. So plastids, they have a structure, again just like mitochondria. They have two membranes, outer membrane and inner membrane. Inside this, there's a fluid-like structure called as stroma. Don't have to remember. Just remember one thing, that they have membranes, stacks of membranes, which are called as, one stack is called as thylakoid, which has pigment called as chlorophyll, and this chlorophyll is helping in absorbing light and is performing the function of photosynthesis. Function of chlorophyll is just to absorb light. So all light dependent reaction which will happen during photosynthesis will be happening inside chloroplast. Where in chloroplast? In thylakoid. Why in thylakoid? Because it has chlorophyll. आएगा break आएगा. Next, other types of plastid, leucoplast. Leucoplast helps in storage. Chloroplast, it's a green pigment and it helps in production of glucose. So chlorophyll is present on the surface of thylakoids. They help in absorbing light energy and hence light dependent reactions takes place here. Same like mitochondria, chloroplast also has their own DNA and ribosome. क्या पता लगता है इससे? Once upon a time, they were also bacteria and some plants grabbed it, said, you stay with us, produce food for us and we'll give you shelter. Same mechanism चल रहा है. Animals ने किसको हड़प लिया है? Energy के लिए माइटोकॉन्ड्रिया को. Plants ने किसने हड़प लिया है? Chloroplast को. That's why they have their own DNA and their own ribosomes. Interestingly, their ribosomes and DNA is similar to bacteria's. Means they were once prokaryotic cell. The ribosomes will be smaller, just like bacteria. Their DNA will be lying without any nucleus, just like bacteria. Next is to give shape to the cell. There are certain fibers or spindles, tubular structures which are spread across. Just like in your body, you have bones to give structured shape to your body. Same way in the cells, there are this fiber-like structure which are spreaded across cell, which maintains the structure of cell and is called as cytoskeleton. Cell ka skeleton, cytoskeleton. Next is, it's a microscopic network of proteins and tubes which provide shapes and coherence to cell. There are filaments, tubes which are present, provides mechanical support, maintains cell shape and helps in movement. Apart from that, cells outer structure may have some other components. For example, you must have felt these days there's lots of pollution and you must be feeling some material lying in your throat when you wake up, right? So that sputum or anything which is there in your throat has accumulated across your day. At night, what your body did was expelling it out, storing it in the throat so that you can remove it in the morning. So what has been done is, interestingly, by the cells of your body, we'll do it again in tissues, but just to explain what is cilia. So your throat, when you did respiration, the air, the polluted air went to lungs. It went to lungs. With air, there were certain dust particles which are going. Now, the cells in this layer has certain hair-like structure. This hair-like structure always propel. They always propel. What they'll do, they'll push the dust outside and they will accumulate here. Same happens when you have cough, same happens when you have any viral or flu, right? So whatsoever respiratory organism which was entering in your body is causing destruction is kept in throat. Why? Because of these fibers like structure which is present here and propelling it outside. And what is these fiber like structures called? Cilia, and it is present on the surface of cell. So cells may also have cilia over their surface, and you know one of the function of them is to move, right? Another structure which a cell can have is, just like sperms have, is flagella, a tail-like structure which helps in locomotion, which helps it to move forward. So there are two structures. Cilia in the respiratory tract is aligned by cells that have cilia, hair-like projections are there, which helps in trapping the inhaled particles. Flagella, it's a tail-like structure which helps in locomotion and movement. Next is some few points, will end this here only, and some few points are that the cells can be, organism can be unicellular and multicellular. If the cell is capable of independent existence, so some organism can remain as a single cell, some may have multiple cells to form a complete individual. Already done that organism can be unicellular or multicellular. Unicellular organism are one celled. Single cell does everything: digestion, excretion, respiration, everything, reproduction. Multicellular, many cells. Cells specialize in their function, and this specialization is seen. Yesterday we have seen most cells, they'll together form tissues, tissues will form organs, and organs will form organ system. Next, there are some entities. We said everything in this world can be living or non-living, but biology is what without exceptions. Exceptions are everywhere. So there are some जो बीच में लटके हैं, complicated relationship. So ना तो इस तरफ है ना तो उस तरफ है, तो ना living है ना non-living है. They are in the middle, right? So जब अकेले हैं तो non-living है, जब किसी host में है तो living है. Same, viruses are one of them. Viruses are made up of genetic material and proteins. They are association of genetic material and proteins. When they are outside the host cell, they are non-living. When they are inside the host cell, they are living, because they need someone else machinery to work. And that's why viruses, can you call them as parasites also? किसी और को feed कर करके जिंदा रह रहे हैं, वही toxic relationship जैसे. So made up of genetic material, show no metabolism of their own, they do not have. And UPSC का favorite है viruses, बहुत पूछता है क्यों नहीं जिंदा, क्या कमी है, जिंदगी में क्या गम खा रहा है इनको, right? तो UPSC का favorite है viruses. कोविड के बाद तो और हो गया. Next, they behave alive only inside the host cell, they cannot be grown in the artificial. UPSC का 2013 में पूछा, फिर 21 में आई गेस दोबारा पूछा कि वायरसेस को ग्रो क्यों नहीं कर सकते हैं या नहीं आर्टिफिशियल मीडियम में. नो, दे नीड समवन एल्स टू फीड अपॉन. तो कोई लिविंग चाहिए उसको, अकेले नहीं. इट कैन नॉट सर्वाइव, इट कैन नॉट बी कल्चर्ड इन एन आर्टिफिशियल सिंथेटिक मीडियम. इट नीड लिविंग सेल्स फॉर देयर culture also. If you want to grow viruses, you need someone cells inside which you'll put virus and that will grow. Next, another two types of complicated relationships are of prion and viron. नाम से ही पता लग रहा है, prions are infectious proteins. Proteins अकेले रह रहे हैं, भटक रहे हैं. They cannot survive on their own, but when they go inside some other body, they can survive, they can multiply also and cause disease. Same virons, infectious, DNA or genetic material. So there can be together genetic material and protein together in viruses. If genetic material is alone, viron. If protein is alone, prion. Simple. Next is if cell wants to die. अब तक तो लाइफ की बात थी, What about death of cell? The cell can die in 3 ways. एक natural death, बूढ़े हो गए, मर गए. एक किसी ने खंजर घोंप दिया तो वो मर गए. एक खुद का मरने का मन था तो मर गए. 3 types of cell death. If it is programmed, if it is natural, for example, programmed cell death is natural death. एक अच्छी उम्र गुजार ली थी, अपना काम कर लिया था और अब मरने गए, मर गए, डेथ हो गई क्योंकि बूढ़े हो गए, ओल्ड हो गए. That programmed cell death is called as apoptosis. Is it good or bad? Definitely good, क्योंकि नेचुरल मैकेनिज्म है ओल्ड वॉर्न आउट सेल को रिप्लेस करने का. सो इट्स अ नेचुरल मैकेनिज्म टू रिप्लेस ओल्ड वॉर्न आउट cells. But if there is some other foreign material or another disease or the cell is not functioning properly, so in that case some external foreign agent can also make cell to die. For example, let's say I got a cut here, so the cells of this region will show inflammation and they will die, so that bacteria and other which are feeding, they cannot survive, or viruses cannot survive. So this will cause inflammation. Such type of cell death is called as necrosis. Okay, UPSC has asked apoptosis, but apart from apoptosis, in that question, particular question, they have given other options also. That's why I have included here. Okay, it's always wise to do other options also, right? कब मन बदल जाए और ऑप्शंस पूछ ले, right? तो necrosis is when there is some foreign agent and cell killing is happening. Now next is खाना नहीं है, कुछ नहीं है, जीना तो चाहते थे बट कंडीशंस नहीं है वैसी, right? तो उस केस में cell, what it does, cell has something called as lysosome. It will break its lysosome and commit suicide, called as autophagy, self-eating. Okay, खाना नहीं है, कुछ नहीं है. For example, when you do not eat food for long time, what happens is, what your body does is, some of the cells will sacrifice themselves, they'll release, they'll break their lysosome and they'll kill their body parts. Again, it will be broken down into carbohydrate, protein, lipids and nucleic acid. Some other cells will feed upon this. So they are feeding upon themselves. Auto means self, phagy means eating, phagocytosis wala phage. So autophagy और suicide. साइड का मीन किसी को पता है? CIDE, साइड. साइड मतलब होता है किलिंग, खुद की किलिंग suicide, पेस्ट की किलिंग पेस्टिसाइड, इंसेक्ट की किलिंग इंसेक्टिसाइड, ह्यूमंस की किलिंग होमिसाइड. Right? Done. So there are 3 types of cell death. First is programmed cell death, which was programmed as it is. It gets rid of damaged cells. It is healthy and expected process. Accidental cell death is necrosis. It is unprogrammed. Any trauma can cause it. Apoptosis, natural. When it is natural, the cell will shrink automatically. It will go. जब बूढ़े होते तो ऐसे ही हो जाते ना, shrink. Same यहाँ पे है. But when necrosis is happening, it will burst. It will swell and burst. Next is autophagy. Autophagy we have done during the times of stress. They eat upon themselves using lysosome. So this is where your cell part gets finished. You want to take break now or after some time? 5 बजे. क्या? स्मॉल ब्रेक. तुम्हारे लिए नया नहीं है ना, औरों के लिए तो है. किन किन के लिए एकदम नया था सेल वाला पार्ट? इट वॉज कम्प्लीटली न्यू फॉर यू. 9th में की थी क्या? Necrosis is when trauma is happening. For example, कोई चोट लग गई, राइट? चोट लगने में वो सेल जो है वो वहां पे मर जाएंगे, क्योंकि उनको आगे स्प्रेड नहीं करना है वो डिजीज या कुछ भी। और कई बार चोट, स्क्रैच लग गया तो ये सेल डेथ है ना, तो चोट लगने पे सेल डेथ होती है, दैट इज नेक्रोसिस। एंड इट इज ड्यू टू एक्सटर्नल एजेंट, नॉट प्रोग्राम। मेरा अंदर से नहीं था कुछ। जब स्ट्रेस है तो अंदर से सेल मर रहे हैं, क्योंकि द कंडीशंस आर लाइक दैट, राइट? प्रोग्राम में नेचुरल होना था, लेकिन किसी एक्सटर्नल ट्रॉमा, चोट लग गई, कोई कट लग गया या एक्सीडेंट हो गया, कुछ भी, उस केस में सेल विल डाई। ओके, बड़े ओए कहाँ निकल रहे हो? नहीं, किसने कहा? ओके, एक काम करते हैं, टिश्यूज की जगह ब्लड ग्रुप एक टॉपिक है छोटा सा, वो कर लेते हैं, उसके बाद ब्रेक लेते हैं, फिर टिश्यूज कर लेंगे। अरे तो मैंने तुम्हारी पहले ही मान ली, मैंने कह दिया कि एक ब्लड ग्रुप वाला टॉपिक जो लास्ट में है वो कर लेते हैं, उसके बाद ब्रेक लेते हैं। इंटरेस्टिंग है, थोड़ा इजी है, राइट? टिश्यूज तो और इजी है, टिश्यूज तो तुम्हें लगेगा क्या ही करा रहे हैं, टिश्यूज बहुत इजी है। जो था यहीं तक था, क्योंकि मुझे ये समझाना था तुम्हें कि बायोटेक्नोलॉजी का बेस कैसे है। वो तुमने ट्रांसक्रिप्शन, ट्रांसलेशन कर लिया, वो सबसे इंपॉर्टेंट पार्ट था, दैट वाज द मोस्ट इंपॉर्टेंट पार्ट, बिकॉज now it will be easy for you to understand what is happening in biotechnology when we are saying DNA is being changed, information is being changed, right? So that was the most important part. पढ़ने यूपीएससी पूछ नहीं रहा है, इट यस यूपीएससी has asked this, but during 90s का जो टाइम है तब क्वेश्चंस थे, तब पूछा है। ट्रांसक्रिप्शन में फ्रॉम डीएनए इट इज कॉपीइंग इनटू आरएनए, बट इट इज नॉट एग्जैक्ट कॉपी। फॉर एग्जाम्पल, तुम्हारी कलर्ड बुक है और मैं जेरोक्स कराऊं, ब्लैक एंड व्हाइट आएगी? है तो कॉपी, बिल्कुल एग्जैक्ट लैंग्वेज सब कुछ सेम है, बस कलर चेंज हो गया। सेम वे इंफॉर्मेशन की एग्जैक्ट कॉपी बन रही है, बट टी की जगह क्या लग रहा है? यू। ट्रांसक्रिप्शन, एग्जैक्ट ट्रांसक्रिप्ट बट स्लाइट चेंज। ट्रांसलेशन में पूरी लैंग्वेज ही चेंज हो रही है, फ्रॉम डीएनए, फ्रॉम न्यूक्लिक एसिड it is changing into protein language, that is amino acid. Okay, it is exactly like British English to American English, ऑलमोस्ट सेम लैंग्वेज है, कुछ अल्फाबेट्स का एस और जेड का फर्क है, बाकी सब सेम है। सो रिप्लेस हो गया, ट्रांसक्रिप्ट बन गया, बट आई ट्रांसलेटेड इनटू हिंदी, जिसके अल्फाबेट से बिल्कुल अलग है, कुछ और ही अल्फाबेट्स है। सो कंप्लीट ट्रांसलेशन चाहिए यहाँ ट्रांसक्रिप्ट से काम नहीं चलेगा, यहाँ कंप्लीट ट्रांसलेशन चाहिए। व्हाई? बिकॉज 26 alphabets and I don't know how many are there are हिंदी। अब सेम लेके चलते हैं डीएनए प्रोटीन पे। डीएनए 4 alphabets, AT GC। RNA 4 alphabets, AU GC। Why we use U? Because it is very unstable, जल्दी मर जाएगा। T is very stable. It will be translated to make, you can make this diagram also for better understanding. Protein which is made up of amino acids. How many amino acids? 20 amino acids. This process is called as transcription. This process is called as translation. कुछ टेस्ट सीरीज में क्वेश्चन भी था बहुत, ट्रांसक्रिप्टोम क्या होता है? अगर एक सेल के, if a cell is working and I take out all the RNA from this cell, RNA has been transcripted. Obviously, any of the RNA must have come from DNA. So all the RNA genetic material is called as transcriptome. Why transcriptome? Because it has come from the process of transcription. So it's the transcriptome. What is transcriptome? After transcription, the cell means I'm not saying for particular gene, but एक सेल ने जो जो इसको काम करना है उसका सारा का सारा जो इंफॉर्मेशन ये आरएनए में कन्वर्ट करता होगा, if I extract that RNA out, that is called as transcriptome. सेल के अंदर हो रहा है सब कुछ, सब कुछ. इट्स अ फंक्शन ऑफ सेल. नथिंग इज हैपनिंग आउटसाइड. ये जो प्रोटीन बनेगा, जो प्रोटीन बनाया राइबोसोम ने, जो भी प्रोटीन बनाया राइबोसोम ने, न्यूक्लियस से इंफॉर्मेशन ली और कोई सेगमेंट आया, दिस आरएनए केम आउट. नाउ यू अंडरस्टैंड द फंक्शनिंग ऑफ न्यूक्लियर पोर आल्सो. बीच में गैप की छोड़े है. इन लोगों को आना जाना भी तो है इंफॉर्मेशन को. कोई मीडिएटर जो है वो आएगा जाएगा, सब काम करेगा. So this RNA will come out. Ribosome will read this information using tRNA. Translator भी चाहिए एक साथ में. Translator के लिए कौन है? tRNA. Because tRNA is the one which can read both languages. एक एंड पे उसके AUGC लगा है, एक एंड पे अमाइनो एसिड की लैंग्वेज रीड करता है. So translator is the one which is reading both the languages. That's why it is called as translator. Okay. So translator RNA, ribosome provides a safe space. So they work together, make the protein. These protein will go to, if it is happening here, this protein will come here. So I said no, ribosomes, they are lying on the ER also. So if this translation process is happening here, so these proteins will be traveling. Some modification if required done by Golgi, and then they can be secreted outside. For example, what your hair follicles are doing. Your hair follicles are making keratin protein. Do this have cell? काटते दर्द नहीं होता ना बालों में. क्यों नहीं होता? Because cell नहीं है. It's just a protein. Your hair follicle has secreted this protein using the same process. ER, Golgi, keratin बनाया और secrete कर दिया. Nails, same process. Okay, so everything is doing the same process. Base यही है. यहीं से पूरी आगे बायोटेक्नोलॉजी चल रही है. Done? Any doubt? Now we'll move on to next topic. That is blood type. How DNA is transferred from parents to offspring? By cells. Eggs and sperm. After fusion, they have nucleus. How endocytosis is different from exocytosis? Endocytosis में अंदर आता है, exocytosis में बाहर जाता है. Entry endo, exit exo. It's all about linguistics. Language समझ लिया, word समझ लिया, means you can crack the meaning of some other words also. Okay. Now moving to blood groups. How blood groups are inherited, or what are blood groups? See, blood contains different types of cells. One of the cell is RBC. Just like we humans have different external appearance due to which we are able to recognize each other. अगर सेम अपीयरेंस होती तो नहीं रिकॉग्नाइज कर पाते, बट ड्यू टू डिफरेंट अपीयरेंसेस, डिफरेंट मार्कर्स, वी कैन रिकॉग्नाइज. अगर आपने किसी को देखा है, उसका स्केच बनवाना है, तो ये बोलते हो ऐसा था, ऐसी आंखें थी, ऐसा चेहरा था. मीन्स दे आर डिफरेंट मार्कर्स. सेम वे over the cell surface there are different markers. There are different markers which are present over the cell surface due to which you are able to recognize them. Okay. Same way RBC के surface पे भी कुछ ऐसे ही markers होते हैं. One of that marker is जो सब में ज्यादातर basically होता है, that is H. Now H modifies itself in different individuals to form A, B, and there is nothing. H के बाद थोड़ा modification हुआ, complete modification नहीं हुआ, तो O. Okay, O here is for 0. कुछ नहीं है यहाँ पे, right? H है, H is the base. There is more modification, A, some other type of modification, B, and modification is half left, O. So, if on the surface of cell there is A, the blood group will be A. If B, blood group will be B. If no such modifications, O. And if it is half left, H. Will do H also at the end. But अभी के लिए समझ लो A, B और O है. Right? यहाँ तक समझ आया? Now, why these blood groups are important? Why? Because inside me, if I have a cell of particular type and someone else, या by other means some other cell of other type comes, then should I interact with it or not? Positive interaction होना चाहिए, negative. For example, you know your friend looks like this. You know the appearance and you know this person is your friend. You'll have positive interaction with it. ठीक है. अब तुमने किसी क्रिमिनल का चेहरा देखा और वो एक दिन सामने आ गया, गले लगा लेंगे. You will not have positive interaction. Common sense में, right? General common sense में you will not have positive interaction with that individual. So we should know that who is positive to us, who is self, and who is non-self. In case of body also, inside the body, cells also distinguish between self and non-self. क्या अपना है, क्या पराया है. जो अपने जैसा दिखता है उसको सही से रखेंगे, पालेंगे, पोसेंगे. जो अपने जैसा नहीं दिखता है उसको, उसके खिलाफ विद्रोह करेंगे, रिवोल्ट करेंगे और उसको किल करेंगे. The same happens in the body. So body also recognizes in self and non-self. To recognize, it needs these markers. So if my body has A blood group, if it has A blood group and B blood group blood is being transfused in me, then my body will react. Why? Because it is not mine. It is non-self. Some other marker, some other person has come and it will react and kill that cells and will lead to inflammation. अगर ज्यादा हो गया तो डेथ भी हो सकती है. Done. Means inside my body, इससे मैं conclusion निकालती हूँ. I get an inference from here that inside my body there are certain army of cells which are working, who distinguish between self and non-self, just like army on the borders distinguish between who is Indian and who is non-Indian and know where to fight, to whom to not to fight. Same, these cells inside my body are also working like that. You call them as immune cells, B cells or T cells. You'll do that in health part. Just know this. And my body produce chemicals called as antibodies to deal with such cells. So if an individual with A blood group is transfused with B blood group, the body will secrete antibodies against B and kill those cells. If an individual is A blood group and A blood group is transfused, so there will be no reaction. अब कुछ ऐसे छुपे रुस्तम जिनका पता ही नहीं लग रहा है कौन, राइट? O blood group cannot recognize, छुपे बैठे हैं, पता नहीं लगता है कौन है. They can also survive because my body cannot recognize it. No recognition means no reaction. So O blood group के कोई reaction नहीं होता because it is not identifiable by the cells, immune cells. Done? Now, if I see an individual of A blood group, on the surface of its RBCs, the marker will be A. Now I am giving a term to this marker, calling it as antigen. This marker is called as antigen. So this marker, A blood group individual will have A antigen over its surface, right? Against whom it will produce antibodies? If it has A antigen, it will produce antibodies against B. So it will have antibody that is anti-B. किसके खिलाफ होगी? B के खिलाफ. If an individual is B blood group, it will have B antigen on its surface and it will produce antibody against A. Done. If an individual is AB blood group, then it is recognized, it is familiar with both A and B. So will it produce any antibody against anything? No. Right? Next is, if an individual does not have A nor B, neither A nor B, any antigen on its surface among AB? No. But it can recognize A and B against itself. So it will have antibody against A and B. Understood this part? Pakka? So if an individual has A blood group, for its familiar part is A, non-familiar part is B, so it will produce antibodies against B. If the individual has blood group B, familiar part is B and non-familiar part is, non-self part is A, it will produce anti-A antibodies. If individual has blood group A and B, it has familiar part both A and B, it will not produce any antibody against A and B. O blood group does not have A, neither A nor B. So since it does not have a familiarity with A and B, so when either A comes, either B comes, it will show reaction because it does not recognize any of this and it will have antibody both A and B. Done. Individual with A blood group can donate to familiar person जिसकी familiarity है A से. individual or the blood groups which has familiarity with A can donate to them. So A has familiarity, even AB has familiarity with A. So A can donate to A can donate to AB. B has familiarity with B also has familiarity with AB. So B can donate to B as well as AB. AB की तो दोनों से familiarity है, but AB के केस में अगर ये किसी को डोनेट करना चाहे तो इसके पास A है जो B के पास नहीं है. It has A which is not with B. So if this individual donates to A, so their blood will has anti-B, they will start killing. And if this individual donate to B, they have anti-A, they will start killing. So they cannot donate neither to A nor to B. What about O? O के पास तो दोनों ही है. नहीं. Means AB can receive from A, from B, from O, but cannot donate. So it is universal recipient. Receive कर सकता है सबसे, दे नहीं सकता. Right? O wants to donate. O does not have any antigen. Now it does not react with somebody else. So O can donate to A, O can donate to B, O can donate to AB, because for O there is no antibody. कोई एंटीबॉडी नहीं है इसके खिलाफ, तो O सबको दे सकता है. जहां भी जाएगा हर कोई खुले दिल से एक्सेप्ट कर लेगा, but no one will react against them. So O can donate to everyone. But यहाँ भी एक गेम है. गेम क्या है? प्लस माइनस का. There is one more marker. आपने ABO का मार्कर तो वेरीफाई कर लिया. One more marker is there, that is RH factor. RBC के ऊपर AB के अलावा एक और फैक्टर है RH फैक्टर. अगर RH फैक्टर है, if there is presence of RH factor, then it is RH positive. If there is no RH factor, then it is RH negative. तो इसका भी देखना पड़ेगा. If I have RH positive, means I'm positive to rhesus factor, RH factor, and the blood of minus comes, my body will not recognize. ले लूंगी आराम से. क्यों? क्योंकि कुछ है ही नहीं ना, पता नहीं लग रहा. रिएक्शन के लिए कुछ पता तो लगना चाहिए ना, सेल्फ नॉन सेल्फ. वो तो मास्क कुछ है ही नहीं, पता ही नहीं लग रहा अपना है पराया क्या है. राइट? लेकिन if I am RH negative and positive blood is transmitted to me, my body will react. दिख रहा है? क्लियर दिख रहा है? मार्कर है तो खिलाफ में काम करना है. So RH negative can donate to positive, but positive cannot donate to. Understood this part? So same way यहाँ पे प्लस माइनस लगा के सेम कॉम्बिनेशन बनेगा. So if individual is A positive, cannot donate to A negative and B individuals also. Done? Understood? Any doubt? Easy? Paka? There is one more marker. For example, if I say the individual has brown hair and blue eyes, तो blue eyes तो देख लिया AB. अब brown hair भी चेक कर लो. तो if it has RH factor, then positive, does not have negative. Negative means कुछ नहीं है उसके सरफेस पे, पता ही नहीं लग रहा है. So if I transmit this blood to an individual, its body cannot recognize this because there is no surface marker. मास्क पहन के, कोई बंदर वाली वो टोपी होती है ना, मंकी कैप, वो पहन के आ गया, पता नहीं लग रहा है अपना पराया है, मारना है, गले लगाना है, करना क्या इसके साथ. राइट? तो doesn't know, we don't know what it is. So our body do not produce any antigen against it. Done? बट पॉजिटिव है if this individual is RH नेगेटिव है, मतलब कि RH को कभी रिकॉग्नाइज नहीं किया, तो जब भी देखेगा लड़ेगा। कभी रिकॉग्नाइज नहीं किया ना, देखा ही नहीं जिंदगी में, कभी घर में घुसा लोगे क्या? जिस अनजान इंसान है, कभी देखा नहीं, ना जान ना पहचान, यू डोंट नो द पर्सन, विल यू अलाउ दैट पर्सन टू स्टे देयर? समझ आया कि नहीं? Our body works on self and non-self. Everybody just think of your blood group. If you are blood group A, बचपन से लेके आज तक 25 साल तक तुम्हारे बॉडी के हर सेल ने, इम्यून सेल ने सिर्फ लेट्स से ए पॉजिटिव, ए को देखा है और RH फैक्टर को देखा है, राइट? सडनली बी कम्स, विल योर बॉडी अलाउ? नो, सो योर बॉडी विल गो अगेंस्ट बी. O comes, but O does not have any marker, cannot recognize. करना क्या इसका? चलो चलते रहने दो, it will accept. Done. समझ आया? जिसका रिकग्निशन है, the one it can recognize it is not mine, it will work against it. One it cannot recognize mine or not mine, self है या non self है, it can escape the system. One it can recognize it is mine, it is similar to mine, it will accept. This is how your bacteria and even viruses work. Viruses also escape immune system. ऐसा क्या होता है कि हमारी पूरी आर्मी सेल्स के अंदर काम कर रही है इम्यून सिस्टम की, then why HIV virus or other viruses are able to enter into cell? Reason is they have certain escape system, means they are non-recognizable. They cannot be recognized by our immune system. आर्मी भी कभी कभी होता है ना, तभी तो घुसपैठी आ जाते हैं अंदर. क्यों? क्योंकि रिकॉग्नाइज नहीं कर पाते हैं अपना है या पराया है. Understood this part? So always, whenever you have doubt, always take upon yourself that my blood group is, let's say, AB. बचपन से आज तक I have seen A also, I have seen B also. So if A comes, I'll accept. If B comes, I'll accept. O में तो कोई recognition ही नहीं है, obviously I'll accept. Done? Any doubt? Any doubt in case of blood group? Done? पक्का? ऑफस्प्रिंग के केस में क्या होता है? मदर का अगर RH पॉजिटिव है, ऑफस्प्रिंग का अगर RH नेगेटिव है, तो ऑफस्प्रिंग विल वर्क अगेंस्ट मदर्स ब्लड. उस केस में कॉम्प्लिकेशंस होती है, राइट? उस केस में कॉम्प्लिकेशन होती है. ऐसा नहीं होता कि स्मूथ चलती है प्रेगनेंसी. ओके, ये केस जो है डिफरेंट है, जहां पे when mother has certain antibodies which can work against the child survival is difficult. Color blindness is genetic, it is not blood group associated. ब्लड का है, मतलब जेनेटिक है, लेकिन ब्लड से एसोसिएटेड है, तो वो ट्रांसमिशन हो रहा है वैसे. ओके, लेकिन कलर ब्लाइंडनेस इज जेनेटिक. डन? एनी डाउट अप टू हियर? बिकॉज आई वांट टू गो ऑन इनहेरिटेंस ऑफ ब्लड ग्रुप. चले? नेक्स्ट इज थोड़ा सा सिंपल करके चलते हैं. अब तक हमने क्या पढ़ा कि न्यूक्लियस के अंदर या हमारे जेनेटिक मटेरियल, आर जेनेटिक मटेरियल अकर्स इन पेयर्स. There are 23 pairs and every chromosome is occurring twice in pair. One pair, one set we get from mother, one set we get from father, right? So chromosome is occurring twice. Let's say the gene on this chromosome is for blood group. The gene on this chromosome we are referring for blood group. On one chromosome, because every chromosome is occurring twice, On one chromosome, the gene says A. The gene says कि ये DNA जिसमें जाएगा, this DNA which will go in anybody's, they will produce A antigen on their RBCs. So it is giving information for A. Other is not giving any information. If such set is occurring in an individual, this individual will have which blood group? A, because O cannot express, O दिख ही नहीं रहा है, पता ही नहीं लग रहा है, so O cannot express, but A can, so individual will have A. Individual will have A because both are having A. So on both the chromosomes, whether from mother, whether from father, he is receiving A, so individual will have A blood group. Individual will have AB blood group. B blood group. Understood this part? Easy. Any doubt? हाँ हाँ हो सकता है, वही बताना मुझे, उसी पे ही आ रहे हैं। सब्र करो। उसी पे आ रहे हैं। Okay. 2 chromosomes in an individual. One has O, another has O. So if I say an individual with A blood group can have A also, combination can be A, AA or AO, means 2 types के क्रोमोसोम हो सकते हैं, A भी हो सकता है और O भी हो सकता है, कैसे भी हो सकता है, means combination can be for A individual, this is also A and this is also A, so there can be 2 different types, AA and AO. 2 different types of chromosome can be there. Possibility is A भी हो सकता है, AO भी हो सकता है, in whole A और O में से कोई तो होगा, right? Next, B के लिए it can be BB, BO, means in whole combination में B और O हो सकता है. Possibility है BB भी हो सकता है, BO भी हो सकता है, एक B तो होगा ही होगा. AB में क्योंकि 2 ही हो सकता है, you have 2 options only, तो A और B ही होगा, there is no chance for O. या तो इस फॉर्मेट में होगा या इस फॉर्मेट में होगा A और B, either deriving A from father, either deriving A from mother, दोनों से A B combination कैसे भी आएगा, it will get A B combination. So the 2 types it will have is A and B. In case of O, nothing, only one type, O. Done. Now let's take an example. Mother, father. Mother has blood group A. Father has blood group B. Combination में genes can be A plus O. It can be anything. It can be AA. It can be O. AO. Right? It can be anything. But in whole A or O की possibility है. I'm just taking possibilities. Just possibility. I just want to know that offspring में कौन सा blood group होगा. What kind of blood group will be there in offspring. Just possibility. That's it. So I can say on the safer side A तो होगा ही होगा, साथ में O भी हो सकता है. B वाले में it can be BB or BO, means combination can be BO, right? Means possibility में क्या है, B और O हो सकता है, उसके genetic material में B भी हो सकता है, O भी हो सकता है. Now offspring की possibility क्या होगी? ऑफस्प्रिंग की पॉसिबिलिटी क्या होगी? ए बी ओ, राइट? समझ आया पॉसिबिलिटीज? हाँ, ए वाला फादर के ओ के साथ जा सकता है कॉम्बिनेशन में, सेल, न्यूक्लियस, एग, स्पर्म, उसका डीएनए, यहाँ पे क्रोमोसोम है, दिस ऑल्सो हैज क्रोमोसोम सेट, लेट्स से दिस क्रोमोसोम सेट इज ए, बट पॉसिबिलिटीज सिंगल ए आया या सिंगल ओ आया या ए आया ओ आया, यहाँ पे पॉसिबिलिटी इज बी और ओ की. This can go with this. What happens is different combinations happen. It is not like both the pairs are shared from mother to offspring, only one set. It can happen if a individual mother has AA. She can give this chromosome also. She can give this chromosome also. Irrespective of which chromosome she gives to child, child will get A. क्योंकि दोनों में A है. But if the combination is AO, child may get this chromosome, child may get this chromosome. Identical होते हैं? Exactly नहीं, variation चाहिए, because we are getting half set of our chromosomes from mother. Okay, simple सोचो. अब मुझे थोड़ा और डेप्थ में जाना पड़ेगा, फिर ये नहीं बोलना कि see what happens is we have 46 chromosomes. Okay, we do not get all the chromosomes, all the 46 chromosomes from both our parents. क्यों? अगर मेरे पास 46 है, फादर के पास 46 है, चाइल्ड विल गेट 92, डबल हो जाएगा. आई विल से ऐसे कैसे? सेम होना चाहिए. ह्यूमंस, एवरी ह्यूमन शुड हैव 46 क्रोमोसोम. आई विल से आई विल गिव माय हाफ ओनली. सो माय 23, टेक एनी 23, बट फ्रॉम पेयर्स. For example, you have 23 pairs of shoes. So from each pair take one. Same way, 23 pairs of chromosome. From each take one, only one. Same way from father side, 23 pairs of chromosome. From each take one. Let's say I have A and A, both of my pairs. But the father has, let's say, BO. On one of his set, there is B. One of his set, it is O. Okay. Because he has also got from his parents. They are not identical. That's why we humans have variations, right? क्यों ऐसा है? Why it is like you are looking in one feature, you are looking similar to your mother, in another feature you are looking similar to your father. For example, your eye color is similar to your mother, but your hair color is similar to your father. Why? Because the chromosome for hair color you have derived from father, but the chromosome for eye color you have taken from mother. And that's why there is lots of variation, and that's why no two siblings are also same unless they are twins, and that too identical twins, right? Because combination can happen in any way, in any way. इतने सारे 23 pairs of chromosome है, कितनी probability है? 2 to the power 23, right? So there is lots of probability, and from other set also it is coming 2 to the power 23. So there is lots of combinations. That's why 7 billion population जो है वो अलग दिख रही है, right? यशराज की मूवी की तरह ऐसा नहीं होता कि जैसे फादर की शक्ल है सेम वैसा ही सब कोई बाहुबली जैसे नहीं है. Because there are lots of, उन्होंने तो पूरा साइंस का कॉन्सेप्ट ही हटा दिया कि सेम फादर जैसे दिखते हैं, मदर के डीएनए का कुछ रोल ही नहीं है। राइट, ऐसा नहीं होता। मदर का डीएनए बोल रहे मैं क्या करूं, मर जाऊं? So what happens is they are combinations. So if we work on probabilities, if an individual is A blood group, he can have A also, AO also. I shortened it for easiness into possibility of A and O. अगर यहां से AO है, AA है, तो A तो होगा ही होगा, AO है तो A तो है ही है, साथ में O की possibility भी है. What I did it, I reduced it for your easiness into these two words. I didn't want this to come. Right? तो possibility क्या है? If an individual, if mother has A blood group, she can have A both on both of the chromosome. In that case also, she has A. If she has A and O, in that case, she definitely has A, but can also have. So this is a probability. And I took two probabilities here. Right? If an individual is B, definitely he has B, one of which will be B. Another can also be B. Another can also be blank, O. So definitely he has B, but another one is the possibility of O. So he has two combinations of B and O. Here also two combinations of A and O. And in any way it can go. Means this A, one from mother, one from father. So I said from A to B, A can go with B also. Blood group will be, A can go with O also. Blood group will be A. O can go with B also. Blood group will be, O can go with, blood group will be. So in this case, if mother has A blood group, or if one parent has A blood group and another parent has B blood group, the possibility is all the four. Understood this part? Any doubt? These all things I've reduced it to this. So take one from here, one from here. Pair one can go with this. It can go with this. It can go with this. Now let's do one more. Let's say parent 1, parent 2, anything, any blood group. Parent 1 has AB. So definitely one will be A, another will be B. There is no chance for O. Parent 2 has O. There is O. Combination एक के साथ हो सकता है. A can go with O and will make A. A can go with O, will make A again. B can go with O. B के साथ कैसे जाएगा भाई? कहाँ है ऑप्शन? पेरेंट 2 के बाद B है ही नहीं. B can go, और let me make it more easy. Probability तो सिर्फ O की है ना? Why take another one? जब A की probability है O, ऑप्शन ही एक है O. किसी के साथ भी जाए A या B ब्लड ग्रुप होगा. The individual cannot have AB blood group. The individual cannot have O blood group. क्योंकि एक पेरेंट के पास क्या है? One parent has AB. At least one it will get from parent 1. A या B में से कुछ तो जाएगा ही, तो A या B का कोई तो ब्लड ग्रुप होगा ही. Done? Understood this part? Now do you know your own blood groups? करो. अपना कोई इमेजिनरी पार्टनर क्रिएट करो, उसका ब्लड ग्रुप सोचो और ऑफस्प्रिंग का ब्लड ग्रुप बताओ क्या होगा. करो. RH पॉजिटिव के अंदर O नेगेटिव डाल रहे हो? हाँ, क्योंकि वो रिकॉग्नाइज नहीं कर पा रहा। नहीं, करो प्रैक्टिस। व्हाट्स योर ब्लड ग्रुप? O पॉजिटिव। संभाल के रखो। कौन सी मूवी में है? अनुष्का शर्मा बोलती है O नेगेटिव, बड़ा रेयर था, है ना? सुल्तान। मूवीज भी सिखाती है साइंस, बाहुबली नहीं। तो मदर के जेनेटिक मटेरियल की कोई वैल्यू ही नहीं रखी। वो भी हो सकता है। कर रहे हो कि नहीं? हो गया? क्या हो सकता है बच्चे का ब्लड ग्रुप? O या A? जो ऑलरेडी है रिलेशनशिप में, उनको तो पता ही होगा ब्लड ग्रुप। कर लो पता। डिस्क्राइब इन द सेंस? मतलब ऑन ऑन ग्राउंड, मतलब जब लैब में आप जाओगे तो कैसे आइडेंटिफाई करते हैं? मतलब जब आप लैब में जाते हो, अपना ब्लड देते हो, आप यही पूछे हो ना कि वो कैसे आइडेंटिफाई करते हैं कि मेरा ब्लड ग्रुप क्या है? हाँ, कि उसके सरफेस पे RH फैक्टर है या नहीं। अगर RH है तो पॉजिटिव, नहीं है तो नेगेटिव। ओके, लैब में कैसे डिटेक्ट करते हैं? वो क्या करते हैं ना, सबकी एंटीबॉडीज डाल देते हैं, जहां से रिएक्शन शो किया, बस वो नहीं है। Okay, for example, what they'll do, if I'll give my blood, let's say I'm A blood group, what they'll do, they'll put NTA, NTB, both. NTB से react किया, B नहीं है, O है. That is what they do. On the slide, they'll put 3 drops, right? One is for NTA, one is for NTB, one is for NTO, NTRH. अगर तीनों में से, let's say, they'll put drops. Let's say it reacted in the first part, जहां NTA डाला था, मतलब A तो नहीं है, it is reacting, B है. Okay, थर्ड वाले में वो RH के लिए था, तो थर्ड वाले में if it is reacting, that means it is negative, क्योंकि पॉजिटिव होता तो रिएक्ट नहीं करता, वो सेम होता. सॉरी, एंटीबॉडी RH की डली थी तो नेगेटिव है. डन. Negative. Definitely both the parents should be RH negative. But again, there is that chromosomal part. Just like O, no? Mother, father. This is positive, this is positive. But it can be negative also. Again, it will show positive only. So same O के जैसे चलेगा, it can be in that case also. Okay. Hidden factor होता है एक. Done? So this is inheritance of blood. The next part is tissue. अब क्या करते हैं, एक ब्रेक लेते हैं. Okay, and then we'll start tissues, but we'll complete tissues part today. और ज्यादा नहीं है, 1-1.5 घंटे का है मुश्किल से. ओके, कितने बजे वापस? वापस का पहले बता के जाओ. 5 5 5. क्या घर जाके सो के आना है? 5:30. 5 o'clock. Okay, now next part of the handout, that is tissues. We have finished cell part. What is the structure of cell? How cell functions? Now we'll see that in multicellular organisms, how cell collaborate together to perform certain functions. So we have already seen the organization that cell together, combination of cell together forms tissues. Tissues together form organ. Organ forms, combination of organs form organ system, and all organ system work in harmony to form organism, right? Same happens here. Now in tissues, so if I say unicellular cell organism, so in a unicellular organism, single cell is performing all the function. There is no specialization. But when group of cells have similar structure or perform similar function, then it is called as tissues. For example, if I say there are WBCs, RBCs, platelets, these all cells are working in harmony. When they are suspended in a fluid, then they perform a function of transport. So it's a tissue. Why? Because it's combination of cell. When somebody says that tissue is formed of similar cells, the statement is wrong. I just gave an example of blood as a tissue which has different types of cells. So when I define tissue, it means either their structure can be same, either their functions can be same or similar, and they are working together, or can be both, same structure and same function. It can also be possible same structure bhi hai, function bhi hai. Structure same hai, ye bhi ho sakta hai. Even only function same hai, similar hai, wo bhi ho sakta hai. Done. So tissue is group of cells having similar structure and or similar functions. Now in case of animals, especially in case of humans, we have 4 types of tissues. When I make a body, when I make an individual body, so if you observe, individual body is like a cylinder. एक सिलेंडर है जिसके अंदर गट है, स्टमक है और फिर इंटेस्टाइन. तो ये भी एक ट्यूब है, उसके ऊपर एक और ट्यूब है. दिस इज वैक्यूम पार्ट फ्रॉम गट till the anus. This is a hollow tube. Outer side is also cylinder. In the middle, all organs are fit together, right? अंदर फूड पाइप है पूरी और बाहर एक सिलेंडर है जिसके बीच में सारे ऑर्गन्स ऑर्गेनाइज्ड है, फिट है, राइट? So these must be made of certain organs and further tissues. Whensoever there is a cavity, any organ, anything, for its protection, does it need certain covering? For example, your body is there. Your body needs covering. You cannot keep everything exposed, right? For example, when you bring in lunch chapati, you cover it with something, right? Sabzi, किसी डब्बे में स्टोर करके लाते हो. Same happens in the body itself. It needs coverings for its protection. So the first tissue here is the epithelial tissue. Tissues, they can be of 4 types. First is epithelial tissue. Thelia means some sheet-like surface. Thelial word is for sheet-like structure. Epi means over something. A sheet which is placed over something. Epithelial means it is covering. So most of the epithelial tissues makes covering protective structure. They are like sheet-like structures, right? Another tissue which you need for your locomotion and movement. When you are moving from one place to another, that is called locomotion. When you are doing movement, not changing the position, that is general movement. For anything of that sort, you need certain tissues which have property of contraction and relaxation. For example, if I am moving my hand like this, so this muscle is contracting because it is reducing in size, and this one is relaxing or stretching, right? So there should be combination of both. And if I am extending my hand, this is stretching and this is contracting. So there should be certain tissue which has this capability of flexibility of contraction and relaxation, and such tissue is muscular tissue, which helps in locomotion and movement. Another tissue which your body needs is to have awareness about your environment. Means if I'm doing like this, so my brain should get the signal that there is certain pressure which is kept on the skin. If temperature changes, anything, sort of pH changes, anything. So my brain should get signals. So everything signaling work should happen so that anything which is happening in the body should be conducted in a proper coordination. For example, if somebody is pressing hard, I should be able to pull my hand back. So my response is dependent on whether I can recognize this or not. For example, sometimes if I'll put finger on your hand like this, you'll feel. When mosquitoes sit, काट के निकल जाएगा, तब पता लगेगा कि बैठा था. Because mosquitoes' weight is very less, you cannot recognize, right? Your body cannot sense that weight. But जब काटा, inflammation हुआ, at that time you'll get to know कि कुछ था, right? Same happens here, that there should be a sensory organ that should be able to transmit all the signals so that control and coordination work can happen, and that tissue is nervous tissue. Now, all the organs of your body or all the systems of your body should be able to connect with each other. Means, अगर कुछ transport होना है, if my gland in brain is releasing some hormone, it should be able to go somewhere, right? If certain other transportation work has to happen, for example, oxygen has to transport from lungs to feet of my body, cells of the feet of my body, then they should be able to transport. So there should be something which is connecting my body parts and functions. If my bone is there and this is another bone, so there should be something which is connecting it, right? So all the connections has to happen. So another tissue which is important is connective tissue. So 4 types of tissues: epithelial tissue for covering, for protection, general. There are other specialized functions also, but I'm not going in that detail right now. We'll go. Another is connective tissue. Main function is structure, support, connections, transport, these. Another is muscular tissue, mainly structure and locomotion and movement. Nervous tissue for transmission of signals. Done. Now let's go in detail one by one for each tissue. First we'll go in epithelial tissue. Now epithelial tissue is the one which is making covering of your body. So epithelial tissue is the one which is making protective lining of your body. Wheresoever a sheet-like structure has to be there for protection, epithelial tissue will be there. Whether it's any organ, whether it's the part of your body, whether it's your mouth lining, whether it is your nasal cavity lining, lungs lining, anything, there should be epithelial tissue. Now, what kind of epithelial tissue will be present depends upon what kind of function you want it to perform. For example, I want a sheet-like structure for protection, but at the same time, I want it so thin that some things can be able to pass through it. So at that time, I want this epithelial tissue should be very thin so that things can transfer from it, or it should work like a sieve for me, that only selected things pass through it. If I want it for protection, I will want it to be very thick, right? So depending upon the function I want it to perform, the sheets can vary. Means these protective linings can vary. I want the sheet that it should be protective also, but at the same time it should be able to handle the stress of acids also. So I will have a lining over it. The sheet will be there, but there will be some mucus lining so that acid does not react to it. So therefore, there can be various types of these sheets. For example, another type of sheet is on your body, skin. You want this sheet that it should be protective also. It should not allow foreign organisms to go inside also. At the same time, you even don't want water to pass through it. Can you absorb water? Can your skin absorb water? अगर हो पाते तो नहाते हुए फूल के निकल के आते, right? नहीं. It cannot absorb water. That's why you have to moisturize it every time to hold moisture, right? हाँ, होता है ऐसे. पानी में पैर रखते हैं, absorb कर लेता ना सारा पानी. नहीं होता ऐसे. Your skin, और एक मिनट, नहाते हुए टॉवल यूज करके सारा हटाते हो ना, you cannot absorb it, वरना टपकता रहेगा पूरा पानी. तो doesn't absorb it, right? हल्का होता है, बट इट डज नॉट एब्जॉर्ब प्रॉपरली कि पानी डाला और यहीं से पी लिया. पीना मुंह से ही पड़ेगा फिर भी, राइट? सो हाइड्रेशन के लिए चाहे कितनी बार मुंह धोते रहो, पानी पीना पड़ेगा. ओके, सो देयरफॉर द फंक्शंस वेरीज. सो एपिथेलियल टिश्यू में दे आर द कवरिंग और प्रोटेक्टिव लाइनिंग्स formed in the animal body. It protects your organs and cavities. What are cavities? I just told you that you have lots of cavities in your body, just like your mouth is a cavity where you put food. The food pipe is a cavity because it's a hollow pipe, right? So those cavities should be lined. So epithelial tissue will be there. This protects deeper layers. For example, your skin protects the deeper tissues present there. Also protect us from injury or infection. Example, lining of stomach. So on stomach there is lining which is again lined by mucus. So if I say this is stomach, so this is the lining, and over this lining there is another layer of mucus which is protecting it, because stomach releases acid, HCl. And if this mucus lining is not there, this will get burnt, and therefore mucus is protecting from it. Okay, so lining of stomach with epithelial cells. It also forms barriers to keep body systems separate. If you want to keep it separate, you will put a lining sheet in the middle. Now the thickness will depend what kind of function you wanted to perform. If you wanted to perform a function where oxygen can transport easily, so you will want this lining to be very thin. And if you don't want any transportation, you want it to be very thick. Permeability of epithelia plays an important role in regulating exchange of material. For example, again, lungs divided into several branches, and at the end you must have heard about alveoli. जब सांस लेते हैं, when you breathe in, oxygen travels from this passage, enters into this windpipe, then enters here, and then there is a bag-like structure which helps in absorption of this oxygen. This oxygen from this sac-like structure, bag-like structure, goes to blood. So if I have to zoom this out and make it here, so there is this bag-like structure, then there is blood vessel. Oxygen from here has to go in the blood because blood is the carrier, RBCs are the carrier, and CO2 from here has to go here, right? This transport has to happen. So in this case, do you want this lining to be very thin? So much thin that it is single celled, matlab just one cell thick. So much thick that just only one cell is present. There should be not multiple layers. No multiple layers, but just one cell thick. Same way, here also you want it to be one cell thick, so that this oxygen has to travel only this, cross this barrier, then this space, and then this barrier. So you want these linings to be thin at certain places and thick at certain other places. Example of thin lining is alveoli in the lungs. In your kidneys also, when filtration is happening, you want that the water should not be released. All the water should not be released. Maximum should be retained by body. So again, these linings help the water to be reabsorbed back. If these linings were thick, water will be drained out of the body. But these linings help that water is again coming in the body and only limited amount of water is expelled out. Understood this part? Any doubt? Next is tissues of small intestine. Another example which help you to absorb nutrients through diffusion and active transport. You have already done what is diffusion. You have already done what is active transport. Urinary waste also get filtered through this epithelia. Skin is another lining of blood vessels, because these are just forming layers. Lung, alveoli, kidney, tubule, everywhere these epithelial tissues are present. Next is they are sheet-like structure. How they are present is, if I have to make epithelial tissue, there will be one layer of base over which cells will stick. So if cell is single layer, there can be single layer also. There can be multiple layers also. Multiple layers will be in the case where more protection is needed. Single layer is where more diffusion has to happen, where you want more diffusion should take place. So it depends upon what kind of function you want it to perform. Now this layer of cells which are present, one important property is they do not have blood supply. They do not have blood supply. That means blood capillaries, blood does not reach them. It remains at lower level, which is called as basement. And properly called as basement membrane or matrix or anything. Okay. Blood does not reach there. So that's why, can I call them, they are avascular. They do not have blood vessels. Lack of vessels make them avascular. A is a term for lacking something, right? So this is avascular. Second thing is that both the ends of the cell are not exposed outside. There is one end of the cell which is exposed outside and another layer, another end which is inside, right? So can I say this is apical side on my skin? This is apical side, and the below which is sticking to that basement membrane is the lower basal part, right? So that is the basal part, this is the apical part, and it is not supplied with blood vessels. अगर ये blood vessel से supply होता, if it would have been supplied with blood vessel, हल्का सा scratch पड़ता, दर्द होता. अभी तो आप कहीं भी टक्कर मारते हो, scratch पड़ता है, white scratch हो जाते हैं, don't get pain much, right? But if it's deeper injury, you get pain, because in that case your pain has reached to the lower levels also, where blood is coming out. Done. Next is then how these cells are surviving, how your skin cells are surviving. If blood is not reaching, nutrients come through blood, oxygen comes through blood, then how these cells are surviving? From the deeper layers, diffusion takes place. From the deeper layer, diffusion takes place. Okay, so directly there is no vessels which are supplying them, but from below, diffusion is helping them to survive, to get food. Done. They are tightly packed, continuous sheets. They have very small amount of cementing material, means within the cell cementing material is less, and almost no intercellular space. What do you mean by intercellular space? Space between two cells. So when the cells are lined next to each other, when cells are lined next to each other, the gap between the cells is almost nil. So there is no intercellular space. All epithelium is usually separated from underlying tissue by extracellular fibrous basement membrane, which we just discussed, that from underlying tissue. So if this is underlying tissue, there is this basement membrane over which epithelium is there. They are avascular in nature, means they do not have blood supply. Blood supply comes from below connective tissue. They are polar in nature, means they have two poles, one side which is exposed to outside environment, one which is not. For example, in my skin, this side is exposed and the below side will be attached to the basement membrane. So that will be basal side and this is apical side. Then is two types of epithelium. One epithelium is there which has general function or protection, which we just discussed. Now certain layers specialize themselves कि हम कुछ secrete भी करेंगे, सिर्फ protection नहीं करेंगे, will produce certain chemicals, certain enzymes and will secrete that also. And these type of cells, what they do is they increase their size, means cell will be there, they'll have proper nucleus, endoplasmic reticulum, everything, and they'll start producing something and will work as gland, means they'll start secreting some chemicals and will work as gland. For example, in your mouth you have lining in your mouth, but जब भी पसंद की कोई चीज देखी मुंह में पानी आया, same happens. Why? Because the lining has also specialized itself to secrete certain chemicals. In this case it is called as saliva, and that lining is called as salivary gland. It is below the tongue, it is round your mouth also. So the same epithelium is working as a gland also to secrete something. Done? Understood up to here? So the epithelium can be glandular in nature, means they can secrete, the cells can work as glands, they can produce certain chemicals and release them outside. Now depending upon where they release their compounds or where they release their components, I can say that glandular epithelium can be of three types. If glandular epithelium releases its product directly into blood, if it releases directly into blood, then such glandular epithelium is called as endocrine glands. Now, blood is transporting across the body, blood is moving across the body. So, if an epithelium, glandular epithelium, which is releasing its chemicals directly into blood, so that chemical can go to any part of your body through blood, means coverage is more. So endocrine glands coverage is more. For example, a gland which is secreting hormones in brain, pituitary gland, can affect your glands at other part also. For example, thyroid gland, adrenal gland, or testis and ovaries also. Means through blood their chemicals can go at larger locations or different locations also. Can your saliva go to different locations? मुंह में रिलीज हुआ था और कल को पता लग रहा है हाथ में था, नहीं, right? ऐसा नहीं होता. Why? Because these are not secreted directly into blood. So where they are secreted? Directly at the site through tubes. So not into blood, but through ducts. Ducts means tubes, and you call them as exocrine gland. You call them as exocrine gland, right? Endocrine directly into, endo means into blood, exocrine not in blood, through duct at the site. Your duct cannot be very long, right? So salivary glands are exocrine gland. Through duct they are releasing saliva into your mouth, not anywhere else. Pituitary gland, thyroid gland, adrenal gland, these glands are releasing their hormones directly into blood and are performing coordination at different sites. Understood up to here? Now there are some glands which perform mixed function. Means they have property of both endocrine and exocrine. Some part of it will perform function as endocrine, they'll behave as endocrine, and some will behave as exocrine. Example is pancreas. Pancreas secrete certain enzymes which are important for digestion. Okay, certain enzymes which are important for digestion. And where does it secrete? Where it should secrete? In blood or in intestine? Intestine, because digestion is happening in intestine. So that part behaves like exocrine. But pancreas at the same time is also releasing certain hormones which are important for blood sugar regulation. Example, insulin. Example, glucagon. These two hormones. Now, what happens is that these hormones has to be directly released in blood, right? So that part is behaving like endocrine. Another examples are testis and ovaries. Testis is releasing hormone like testosterone directly into blood. So there it is endocrine. It is also releasing sperm into sperm duct. There it is exocrine. Ovaries release hormones like progesterone directly into blood, endocrine. It releases eggs into fallopian tube, into a duct. There it is exocrine. So there can be, when there are two functions mixed, you call them as heterocrine gland. So glands can be of three types: exocrine, endocrine, and heterocrine. Done. So glandular epithelium, they function by releasing, secreting certain substances. They can be of different kinds. First kind can be endocrine, that releases hormones directly into your bloodstream. These glands act like chemical messenger, because message from one part of the body is transferred to the other part of the body. So message can be transferred in two ways. First way is through nervous system. Another way is through chemicals. For example, if my brain has to tell my body that release thyroid hormone, right? So thyroid gland जो है, pituitary gland from the brain will release hormone which will via blood will go to thyroid and hit it, activate it, that release thyroxine, release. And again, if it does not, you'll feel like which disease? आयोडीन की deficiency से होती है goiter, right? Suppose brain is again and again signaling your thyroid gland that secrete thyroxine. Thyroid gland wants to secrete, but it does not have iodine. Again and again brain is pushing it. तुम्हें कोई बार बार पोक कर रहा है, काम कर कर कर, और तुम्हारे पास बुक ही नहीं है उसकी, तुम्हें क्या आएगा? मुंह फूल जाएगा, सूज जाएगा. Same happens to thyroid. Inflammation happens and you face goiter like disease. Done. Pancreas role is twice. Two roles are there. First is digestion. Mein digestion, okay, just when you eat food, it goes through stomach, in intestine, it has to be digested, right? Specific part of digestion is here. Here is pancreas, a leaf-like structure. This pancreas releases certain enzymes. I just said in the previous class, enzymes are certain proteins or chemicals which helps in breakdown. Chemical reaction करवाते हैं ये. So if I have to digest, carbohydrates will digest into glucose-like structure. Proteins into हर जगह सेम चल रहा है, पता है? सेल में जो किया वो हर जगह सेम है. Proteins into amino acid, carbohydrates into glucose or monosaccharides, fats into lipids, lipids into fatty acids and glycerol, etc. And nucleic acids into nucleosides, a different property. So same chemicals are here also, and pancreas secrete them, and due to which digestion happens here. Done. Another function of pancreas is to release hormones, right? And two hormones particularly, insulin and glucagon. Both helps in blood sugar regulation, one by lowering it, one by increasing it. So if your blood sugar level, you just ate sugar or some sweet, your blood में sugar जो है बहुत ज़्यादा है, it has spiked. So this insulin will start releasing, pancreas will start releasing this insulin. This insulin will go to cells and ask the cells that take up this sugar and store it. So cells will start storing the sugar from the blood and blood sugar levels goes down. Now let's say you didn't had food for a long time. In that case, glucagon will do reverse. Glucagon will help that sugar come out in your blood. Done. Means it is not working. Insulin is there, but still cells are not taking. Resistance में ढीठ हो गए जैसे कि पढ़ने को बोल दिया फिर भी नहीं कर रहे हैं. तो same way insulin resistance. Okay, it is not working. Done. Next, thyroid gland secretes thyroxine hormone. What's the function? Metabolism. Metabolism. अगर किसी की body ज़्यादा गर्म रहती है, if someone's body is remaining for higher temperatures or above average body temperature, at that time they usually say कि thyroid बहुत ज़्यादा अच्छा होगा, जिसमें हम देसी भाषा में बोलते हैं metabolism बड़ा अच्छा होगा इनका. What they actually means is कि thyroid hormone थोड़ा ज़्यादा अच्छा होगा. और जब वेट बढ़ने लगता है, we say कि thyroid चेक करा लो, हो सकता है हार्मोन कम हो. Means मेटाबॉलिज्म कम हो रहा है, तो स्टोरेज हो रहा है सब चीज का, बर्न आउट नहीं हो रहा है, कंबशन नहीं हो रहा है फूड का. सो व्हाट हैपेंस इज अगेन मेटाबॉलिज्म. इन केस ऑफ मेटाबॉलिज्म, थायराइड ग्लैंड वर्क्स. Next is pituitary gland. It is called as master of all gland because it controls all the gland. They are examples of epithelial glands only, glandular epithelium. Next, adrenal gland, fight and flight hormones. In case if you see a dog barking at you, adrenal gland will secrete hormones, adrenaline, noradrenaline, which will help you to deal with that condition. Next is exocrine gland, which secrete their juices or their secretion directly into tubes or ducts. Example is sweat gland in your skin. Sweat glands are there which are releasing directly into duct and it is coming out. मूवी टीवी एडवर्टीजमेंट में जब वो दिखाएंगे ऐड तो ऐसे डक्ट बना के दिखाएंगे एंड द केमिकल इज कमिंग आउट, राइट? सो दीज आर दोज डक्ट्स. नेक्स्ट, सलाइवा, salivary glands, mucus in the respiratory tract. You have mucus here. We just discussed that at the morning time you'll feel sputum because of that epithelium. Next, stomach acids, again epithelium is there and it is releasing mucus. Milk mammary glands, again example of exocrine gland which is releasing milk. Next is sebum oil gland, your skin. किसी की oily skin है, किसी की dry skin है. So oily skin is because your oil glands are releasing that oil. Sebum. Next is heterocrine glands. Heterocrine glands are mix of both. Means some part of it will work for as exocrine and some will work as endocrine. For example is pancreas, unique dual function. both exocrine and endocrine. 95% of it is exocrine, means it will release enzymes directly into intestine, and 5% of it is endocrine, it will release directly a hormone into blood. Two hormones, glucagon and insulin. Next is gonads, that is in males it is testes, in females it is ovaries. In females they are releasing estrogen and progesterone hormone, and in males it is testosterone hormone. Next is connective. So epithelial tissue is ended. Next tissue is connective tissue. Now connective tissue is quite different from epithelial tissue in the way of its structure. Connective tissue is very diverse. Diverse in the sense it can be hard, it can be flexible, it can be fluid also. For example, blood. Blood is fluid in nature. So on the hardness scale, if I have to measure, it can be as hard as bone, it can be as non-hard or pliable or fluid as blood. So there is lots of diversity in connective tissue. Now how these connective tissue are formed? Just imagine there is a jelly, and in that jelly you have put gems. Now this jelly is like matrix like structure, and those gems are like cells. How much soever cells you put, it will become more hard, and more matrix, more hard. So hardness will increase. If this jelly, you make it very fluid, flowy, and the cells are scattered around it will become more fluid in nature. Example, blood. So connective tissue is made up of two things, matrix and the cells. Where is this matrix? The matrix is lying outside cells. So let's say this is cell and the matrix is here, all around it. And apart from that, there can be fibers also. So if more fibers are there, more stiffness, less fibers, less stiffness. For example, in blood, fibers are not there, fluid, it is flowing. But in case of bones, cartilage, there is fibers, collagen is there, lots of things are there, cells are there, densely packed. So means matrix का में cells, fibers is पूरा densely packed हो गया and it is hard in nature. So flexibility scale पे there is lots of variation. How they are formed? Two things, matrix and cells. Connective tissue is almost everywhere. The most largest part of this tissue chapter is connective tissue. Means almost 50% of time we are going to devote to connective tissue. So connective tissue is almost everywhere. It depends upon how much it is there. It depends upon tissue to tissue. Example, skin has mostly connective tissue because below the skin there is lots of connective tissue. Why connective tissue? Because it has to attach with the muscles. It has to attach with bones. तो जितने सारे organs हैं सबको आपस में connection करना है. Connective tissue almost ज़्यादा ही होगा. Skin को attach करना है muscle से, connective tissue. Muscle को attach करना है bone से, connective tissue. एक bone को दूसरी bone से attach करना है, connective tissue. Bone अपने आप में connective tissue है. Okay, because it is connected. Blood is another connective tissue. They are named connective tissue because they have special function of linking and supporting other tissues. They range from in softness from hard to soft. They are made up of two things, matrix and cells. Where is the matrix lying? Outside cell. So it can be called as extracellular matrix because it is lying outside cell. Can you say it is lying between two cells? Yes. Can you say it is intercellular matrix also? So the same matrix can be called as extracellular. It can also be called as intercellular, not intracellular, intercellular. दो cells के बीच में. The matrix can be jelly-like, fluid, dense, rigid, jelly like भी हो सकती है जैसे एल्वियोलर टिश्यू में होती है, फ्लूइड हो सकती है जैसे ब्लड में होती है, dense और rigid जैसे bones, cartilage इन सब में होती है. In all connective tissue except blood, cells also secrete fibers which maintains their proper structure, and these fibers are called as collagen or elastin. Next is ये जो extracellular matrix है, दो चीजें हैं इसके अंदर. We have made two things. One is that jelly-like structure and then those fibers. So there is ground substance which has this watery rubbery texture and there are fibers scattered along it. उसी के अंदर cells भी है. So it has ground substance as watery rubbery unstructured material. They'll have lots of nutrients, starch, protein सब यहाँ होगा. Fibers में there'll be fibers there, example collagen fiber, and they'll help to make structure of it. Types of connective tissue based on क्या density है. Connective tissue can be of two types. Majorly हम तीन करेंगे यहाँ पे, but density के basis पे connective tissue can be loose connective tissue. Loose means ज़्यादा dense नहीं है, proper structure shape नहीं है. It doesn't have proper structure and shape, but it is loosely found. And dense connective tissue which has a proper structure shape because of the density it has got due to large number of fibers, thickness, cells present there. Another category just for better understanding we have made is specialized which perform special function. So first is loose connective tissue. First type of loose connective tissue is wheresoever vacuum space, vacant space is there. So you want to fill that part. For example, when you store something in a box, packaging में आजकल आता है and they wrap it, right? So, and if you want that this thing should not be damaged, so you'll protect it by filling it with cotton or something. So wheresoever vacuum space is there our body has filled it with a alveolar tissue. जहाँ पे कुछ भी space है तो वहाँ पे present है. Areolar tissue वहाँ पे present है. मतलब जहाँ भी gap आ गया, wheresoever gap is there, wheresoever vacuum space is there, just for stuffing, just for filling, areolar tissue is present. It is loose packaging tissue found under the skin or muscle, around blood vessels, nerves, जहाँ भी space है वहाँ पे fill हो जाएगा. Another type of tissue is adipose tissue which stores fat and is usually found under the skin. Adipose tissue are fat storing cells found under the skin between internal organs and they store lots of fat inside them. They have fat globules. Example structure is cell, nucleus गया side में. Here there will be lots of fat. And this is adipose tissue. Areolar tissue will support internal organs. Adipose tissue भी वो काम कर सकता है, but main function is store of fat and heat storage. Okay, so it works like insulator and doesn't allow heat to transmit outside. Next is dense connective tissue. In dense connective tissue, the first tissue is tendons. See, connection जो है skin, muscle, then bone. This muscle should be attached to bone and there should be some connective tissue which is attaching that muscle to bone. For example, अगर ये stick है, I have to paste something over it. So in the middle, there should be some tissue which should connect it with that surface. Here it is tendon. What's the function of tendon is to connect a bone with the muscle. So if you have this hand bone humerus and there are biceps and tricep muscles, So the similar way you have their tendons in the middle of that, which is attaching that bone to the muscle. So when your hand is moving, simultaneously that bone and muscle are interacting. So what happens is, from muscle to bone, or you can say reverse, bone to muscle, which tissue is helping? Tendon is helping. Now, it is fibrous tissue, strength है, तभी तो movement हो पा रही है. अगर नहीं होता, if it wouldn't have strength, any strong movement would you have done? It would have broken. Next is bone to bone. So if one bone is there, you need to connect it with other bone, right? So there should be certain tissue around it, and that tissue is called as ligament. Ligament में fracture होता है, वो भी टूट सकता है, right? So that tissue is called as ligament. So one bone, this bone is attached to this bone using a ligament. This ligament should be rigid or flexible. Flexible तभी तो joints में movement कर पाओगे. अच्छा, bone और bone के बीच में space छोड़ा क्यों है? एक ही bone क्यों नहीं डाल दी है? Movement नहीं होगी. Joint किस लिए चाहिए? Movements के लिए चाहिए, right? और वो joint पे ligament भी चाहिए ताकि वो दोनों bone connected रहे. So you need two bones, you need a breakage for movement, and you need a ligament to stay them or to make them connected. It is just like door, उसमें hinge भी चाहिए, right? So tendons, then ligaments, they attach bone to bone. They are very elastic. That's why your movement is possible. Ligament contains very little matrix. Next is specialized connective tissue. Specialized connective tissue, the first is blood. Blood में, again I said, connective tissue में there is one matrix kind of structure and there is cells which are present. So matrix kind of structure here is called as plasma. Cells are there separately suspended within the plasma. Cells are of different types. RBCs are there who has hemoglobin, perform the function of transporting oxygen. Cells, does RBCs transport carbon dioxide also? सिर्फ ऑक्सीजन बोलते हैं, कार्बन डाइऑक्साइड नहीं करता क्या? नहीं करता. ब्लड में कार्बन डाइऑक्साइड ट्रांसपोर्ट होती है बट नॉट विद द हेल्प ऑफ आरबीसीज. प्लाज्मा में डिसॉल्व होकर डिफ्यूज होकर होती है. दैट्स व्हाई बचपन से रटते आए हो, हीमोग्लोबिन ट्रांसपोर्ट ऑक्सीजन, ऑक्सीजन ही है. जब लंग्स के पास जाता है तो ऑक्सीजन आ रहा है, कार्बन डाइऑक्साइड जा रहा है, तो वो कहां से जा रहा है? ब्लड से जा रहा है, नॉट फ्रॉम आरबीसीज. डन. देन अनदर टाइप ऑफ सेल्स इज WBCs. What is the function of WBCs? Defense, immunity, fight the foreign agents. Another type of cells is, they are not particularly cells, they are fragment of cells. What is their function? Coagulation of blood. What does that mean? What does that mean? Prevent loss of blood during an injury. So if you get cut, any injury, if there is loss of blood, so platelets will come there, they'll release certain fibers, they'll plug that area so that no more blood loss is there. If this condition cannot happen, if this coagulation does not happen, what does that disease called as? Hemophilia. So hemophilia is a disease where coagulation of blood cannot happen. And it was famously transmitted, or most popularly in which family it was there? Royal family of Britain. Even in Russia, because they married within their families. So it was most popularly transmitted, or because it's a genetic disorder, so it was transmitted across generations in the royal family of Britain. Done. Next is plasma. Now this plasma is a straw-like, colored-like structure, fluid, slightly yellowish, and it contains water, it contains hormones, it contains salts, sugars, everything which can be dissolved in water. So all the hormones which we just studied, when they are transported through blood, how they are dissolving? They are going through plasma. They are not entering into blood cells. They are going through plasma. बह के जा रहे हैं. So blood is a fluid liquid matrix called as, it has plasma in which RBCs, WBCs and platelets are suspended. Plasma contains protein, hormones, salts. Blood flows and transport gases, digested food, hormones, everything is transported through blood. So if you are eating something, digestion is happening inside the intestine. So from intestine, it is reaching to your muscle cell through blood. Blood is absorbing it, taking it to the muscle, take the food, blood, RBCs are taking oxygen from the lungs, take the oxygen, burn it, use it for locomotion and movement. Done. So they are getting the energy there. Blood is taking food, plasma is taking food, and RBCs are taking oxygen. Done. Any doubt? Next is bones. Bones are strong, inflexible tissues. They are non-pliable. Non-pliable means stretchability, flexibility. Nahi hai, they do not have much flexibility. They are rigid, made up of calcium and phosphorus salts. Apart from that, collagen fibers are there, which gives bone the strength. Structural framework जो body का बनाते हैं. Why you are not like amoeba? बह क्यों नहीं रहे हो? Why you have a fixed structure is because of this skeletal system, that is bones. Bones support and protect softer tissues and organs. Limb bones such as long bones of legs serve weight bearing function. इसके अलावा bone एक और function करता है, that is production of RBCs, WBCs, blood cells. तो bone के अंदर there is bone marrow, and in bone marrow blood cells are produced. Next is cartilage, the softer version of bone. So cartilage is slightly more flexible. It is present at the tip of your nose, your ear. So cartilage जहाँ flexibility चाहिए वहाँ पे cartilage present है. It is slightly more flexible than bones. Another type of connective tissue is cartilage, widely spread, solid matrix है, proteins और sugars का बना है. Intercellular matrix of cartilage is solid, pliable. Bone का क्या था? Non-pliable means not flexible. Here it is flexible and resist compression. What is the role of flexibility with compression? अगर hard होगा, if it will be hard, I'll put anything, any compression, it will break. If it is stretchable, flexible, it will cushion, it will just press and come back again. So cartilage is needed there. Cartilage is present at the tip of your nose, outer ear joints, between adjacent bones, vertebral column में present है. Cartilage also smoothens bone surface. Wherever bone surface is hard, rigid, it smoothens it out. What is the function? जितने भी, whatsoever we have done till now, each individual type of connective tissue is the function. Storage भी function है, support भी function है, transport भी function है. So connective tissue is used in all these things. One thing that is common in all connective tissue is, first thing, they have common origin. What makes them different from other tissues? Muscular, epithelial. First, they are made from mesenchyme. Same. Degree of vascularity is variable, or blood flow. Cartilage are avascular in nature. कोई blood vessel नहीं है. कई जगह बहुत ज्यादा blood vessel है. Blood तो अपने आप में ये connective tissue है. Mostly composed of non-living material, while muscular tissue, lots of cells, epithelial tissue, lots of cells, nervous tissue, cells. So non-living material is more in connective tissue as compared to other tissues. Next is muscle tissue. अब देख के हिम्मत बढ़ रही होगी, खत्म हो रहा है ना? तुम्हारी क्या, मेरी भी बढ़ रही है, हो रहा है मुझे बहुत तरस आता है जब इतना ज्यादा तुम लोग ऐसे बैठते हो ना हाँ, कोई बोल रहा है ना कुछ? नेक्स्ट इज muscular tissue. So muscular tissue has contractile nature, means it can contract and relax, it can stretch, and it helps in locomotion and movement. Now muscular tissue is highly vascularized, matlab there is lots of blood supply there. Why blood supply? Because food is needed, oxygen is needed. When it is working, it is producing carbon dioxide, so that has to be taken back to lungs so that it can be breathed out. So lots of vascularity is there. Why? Because blood vessels are required for their proper functioning. So it is highly vascularized. It has proper nervous tissue attachment, means lots of nerves are connected so that they can work in coordination with each other. If I'm giving a signal, I have to pull my hand. So this signal has to be transported and nervous tissue should be well coordinated and integrated with this system. So nervous tissue integration hai, vascularization hai, and there is a property of stretchability, that is contraction and relaxation, which we call as contractibility. Next, 3 type के muscle tissue. There are 3 types of muscles. One which is striated. Striated means that when you see them under the microscope, you will see certain band-like structures, striations, which are called as band-like structures. You can call them as dark band, light band. Why? Because this will appear dark, this will appear light. So there are striations which can be seen under the microscope. These muscles have multiple nucleus, means the cells of these muscles will have multiple nucleus. If multiple nucleus are there, what does this mean? We have already done this. Lot of functions, lot of work has to be done. Next, these muscles help you to perform your voluntary task. What do you mean by voluntary task? The tasks which are in your own control. For example, moving my hand from here to here is in my control. So intentionally I am doing it. So that means this is done by striated muscles. Signals are reaching there and movement is happening upon my wish. So these are voluntary in nature, multinucleate, and striations are seen on its surface, where it will be present according to your bone skeletal system, because skeletal system, most part of it is under your control. So all the movement, locomotion which you are doing is under your control, and that's why, since these muscles are associated with skeleton, these muscles are also called as skeletal muscles. Done? Easy. Next is another type of muscle. If I see it under the microscope, I do not see striations, so I can call it as non-striated या unstriated. So how do they look like if they do not have striations? They look like spindles. They look like spindles. Spindles is something which is broader in the center and tapering at the edges. They have single nucleus, uninucleate, And these muscles are non-voluntary. These muscles are non-voluntary because that means that they are not under your control. So how the work is happening if they are not under your control? भगवान भरोसे भगवान भरोसे चल रहा है, अपने आप चल रहा है ऑटोमेटिकली. फॉर एग्जाम्पल व्हेन यू ईट फूड, यहां तक तो खा लिया, गटक लिया, उसके बाद द फूड इज मूविंग ऑन इट्स ओन. यू आर नॉट सेइंग नीचे जा नीचे जा नीचे जा या फिर pushing it, right? So it is on its own. Food pipe is taking the food downwards. How it happens is the food came, there will be contraction here, food will be pushed down. When food came here, there will be contraction here, food will be pushed down. So in the pipe, these movements are happening: contraction, relaxation, contraction, relaxation, contraction, relaxation. Particularly called as peristalsis, don't have to remember. So due to this movement, food is automatically going down. When someone put torch on your eye, automatically the pupil compresses, the muscles of iris are stretching so that light does not enter, right? So again, that is not under your control. So there are lots of functions which are not under your control. They are automatically happening due to smooth muscles. Done. Next is the third type of muscle which is present, and that is again not under your control. That is cardiac muscles. They are cylindrical, just like striated muscles. They are cylindrical muscles, just like striated muscles. Just like smooth muscles, they are non-voluntary or involuntary. दिल को कंट्रोल में रख के नहीं बैठे हो, आज सुसाइड करना है तो मन मर्जी से दिल की धड़कन रोक देते हैं, नहीं. यार दिल धड़कता रहता है, कितना ही दिल टूटा हो तो उसको कंट्रोल नहीं कह सकता. यू कैन नॉट कंट्रोल दी movement or contraction relaxation of cardiac muscles. So cardiac muscles are involuntary muscle. They are cylindrical. They have properties of both the types of muscle. They are cylindrical, but at the same time they are involuntary in nature. Muscular tissue done. So muscles are of three types, usually based on the structure, how they are seen. Two types: voluntary muscles or skeletal muscles. They can be moved at conscious will, mostly attached to your skeletal system. They are striated, means striations can be seen when you see them under the microscope. They are bundled together vertically, means cylindrical bands are there. Alternate dark and light band are there. When you make striations, you are making dark band, but the alternate light band is also there. They are called striated due to presence of these striations. Multinucleate. So if you see, there is alternate dark and light band. There are multiple nucleuses here. Striations are there and they are voluntary in nature. They are spindle shaped. They are uninucleate. They are not in your control. Again, cylindrical striations are there. The junction between adjacent cell. Coordination is more so that the complete heart contracts together and relax together. Okay, the chambers of the heart. It should not be like few of the cells of the heart are contracting and few are relaxing. It happens, but according to chambers. Next is smooth muscles. They are tapering at the edges, at the ends. They are uninucleate, unstriated muscles. Example, alimentary canal. What is alimentary canal? From food to the gut system, from food to anus, that is alimentary canal. That is one tube which has different variations. Contraction, relaxation of blood vessels, iris of eye, bronchi of lungs. Cardiac muscles, they are cylindrical, branched, striated and uninucleate. Again, they are not in your control. Communication junctions are there so that they can work together as a unit. Muscle of heart has rhythmic contraction and relaxation, and it can be found in next, and the last is nervous tissue. Now for nervous tissue, the fundamental unit is neuron, the cell which helps in transmission of signal from one end to another end. What is the end? There is this spherical body at one end which has proper nucleus, ribosomes, mitochondria, etc. Then there is a tubular structure which further divides into another structure and end into this bulbous or bulb-like structures. This is called as cell body or soma. Don't have to remember it. Cell body is enough, which has nucleus. This is called as a long tubular structure is called as axons. Here they are fibrous structures which are called as dendrites. Dendrites is anything which has lots of fragments, edges, just like your tree branches. So lots of branches is there, then it is dendritic structure. Signal moves from here, from this direction to this direction, from cell body to axon endings or nerve endings. Here, if there is a muscle, it will transmit signal to muscle. For example, I pressed here, pinched here, this signal has to reach to my brain. So there should be some nerve which is carrying that signal. So there should be some neuron which is carrying that signal. From the brain it has processed, and another neuron is carrying a signal which is giving to my muscle that pull your hand back. So that signal will be transmitted from neuron in my brain, and slowly it will go to muscle, and when the muscle will get the signal, it will pull the hand back. It is happening within seconds and microseconds. So the complete reaction is very fast. Done. So the signals can be reached to muscle. Now how the signal will be communicating or traveling? Here the signal is moving as electrical impulse by movement of ions. Yesterday we have done active transport, sodium and potassium pumps. So it is traveling by sodium and potassium, concentration changes and electrical impulses generated. Here there is space. Circuit खुल गया. Electrical वाले जानते होंगे, circuit should be closed for electrical impulse. Now here it will move through chemical called as neurotransmitter. सुना होगा. जब पेन किलर लेते हैं तो क्या होता है? When you take painkillers, suppose you have pain in your leg or a muscle, you take painkiller, right? Why? Because that neuron is giving you signal again and again to your brain that something is paining there. What you did, you didn't cure the root cause why it is paining. You stopped the signal. जब गर्लफ्रेंड बार बार परेशान करे, फोन ही स्विच ऑफ करके रख दो, सिग्नल कम्युनिकेशन ही बंद हो गया. Same happens here, that it is giving signal. Stop that neurotransmitter, block कर दो जहां से सिग्नल ही उठा रहा है. मतलब जो दवाई आपने खाई, एस्पिरिन, डिस्प्रिन, it will go there, it will stick to that site so that neurotransmitter does not stick and give signal. Okay, या तो फोन बिजी रखो या फिर बंद करके रख दो. It happens here also. So this is how your medicines are working. This is how your signal is moving. So nervous tissue, All the cells have the ability to respond to stimulus. Living individuals should have ability to respond to stimulus. Cells are highly specialized. They are being stimulated. जब भी stimulus मिलता है, they get stimulated and send the signals. Neural tissues have greatest control over responsiveness because it happens in very quick moment. Their reach is high. Big functions are sensing stimuli, sending electrical impulse through the body. So it has to sense also. It has to release signal to the muscle for the reaction also. So sensory part also, and reacting part also, and processing part also. 3 functions has to act simultaneously. Done? Next, what are the sensory part, or what are the processing parts? Brain, spinal cord, and nerves are there. Nerves are for transmission. Brain and spinal cord is for processing. Nervous tissue is made up of 2 types of cell. First type of cell is neuron. Neuron contains a cytoplasmic cell body which also has nucleus and hair-like structure which are called as dendrites. These dendrites are the ones which are carrying the signal to the cell body, and then through axon it is being transmitted. The cell body is called as soma, which performs all the functions which are important for sustaining the life of nucleus. It has mitochondria, everything which we have done in cellular part. Dendrites, they collect the signal. Axon, they transmit, and they are present all over the body. When a neuron is suitably stimulated, what do you mean by suitably stimulated? अभी बोला मस्किटो बैठेगा तो सिग्नल नहीं जाएगा, उतना थ्रेशोल्ड का सिग्नल भी होना चाहिए. So that threshold signal should be there, then only it will be transmitted. Then there is another cells which are supporting the functioning of neurons, that is glial cells. एसी चल रहा है? पता नहीं चल रहा, गर्मी तुम लोगों को भी लग रही है? Next is plant tissues. Now plant tissues, again plants have multiple cells, organize themselves into tissues that perform certain function. There can be different structures for different tissues. There can be different functions which they perform. Now type of plant tissues can be, first, if they are growing continuously, means they are at growth stage. They are dividing and not performing the much specialized function, but they are continuously dividing. Then they are called as meristematic tissue. So if they are continuously dividing, their role will be more in growth. Their role will be more in growth. So if I have to say, where should be these meristematic tissues present? Should it be present here at the tip for the growth? Should it be present at the lateral side for the growth of girth? Should it be present between the branches? So there are 3 locations. First is tips, that is apical part. Lateral on the sides for lateral growth, that is lateral meristematic tissue. And between the branches, that is intercalary tissue. Easy. Next is permanent, means they are not growing now, but are performing specialized function. Are performing specialized function. Now, based on whether they have single type of cells, they can be simple permanent tissue. Whether they have multiple types associated, they can be complex. Done. Example of simple permanent tissue is parenchyma. Single type of cells are there and they are performing the function of photosynthesis or storing water or air, anything, right? Complex mein the example is xylem and phloem. What is the function of xylem? Transporting water and minerals करेगा कि नहीं? वाटर के साथ ही बह के जाएंगे सब, तो वाटर एंड minerals, phloem transporting food. Okay, so this is the complete chart of plant tissues. Plant tissues can be, if they are continuously dividing, meristematic. If they are lost the division property, specialized, permanent, made up of single type of cells, simple, multiple types of cells, complex, xylem and किसके? Parenchyma. Parenchyma. Chyma means a mass of cells. So plant tissues, just like animals, even your plants has collective tissues which are made up of several types of cells. They are very different according to their environment, different structures. Generally, they are supportive in nature. For example, parenchyma, collenchyma, they support the structure of plant. Next is, most of these tissue can be dead. It's not like they get separated. Sometimes few of the tissues get separated. For example, leaf during autumn, they'll get separated. Plants are very unique and I would say more advanced than us. हमारा कोई organ खराब हो जाता है तो हम उसे intentionally तोड़ के गिरा नहीं सकते. But plants, every year they'll get new leaves and leaves will fall depending upon what they want it or not, right? So here also they can have dead tissues attached to them, for example, bark of stem, right? Different functions. Plants are again very different from animals because they are stationary, they have to tackle with extreme conditions, unlike us. So they have protective tissues always attached to them. So different functions are performed. Meristematic tissue always growing, permanent tissue, they are not growing. Animal tissue mein there is no such demarcation that is dividing and it is non-dividing, but in case of plant it is there. Types of plant tissue. First, meristematic tissue, certain specific condition, they are always growing. What are those conditions? Apical, intercalary and lateral meristem. Again growth, apical lengthwise growth करेगा, lateral girthwise, intercalary between the branches. Next, these cells are very active. Why? Because they are rapidly growing. They have dense cytoplasm. Why their cytoplasm will be dense? Thin cellulose walls and prominent nuclei. All the three things goes to one point. Why? The answer for all the three things is same. Because they are rapidly growing. अगर rapidly growing है तो हो क्या रहा है? If the cell is there, nucleus is there, I keep it to rest. Means I say don't grow. So its cytoplasm will keep on increasing. But there is another cell, I divide it, immediately it becomes small. Nucleus is clearly visible. Cytoplasm became more dense. Now again it is again dividing, more prominent. So in such cells, because they are growing, they are not getting time to increase cytoplasm, due to which their nucleus is very prominently visible. I am not giving it time to develop cellulose wall to harden also. That's why cellulose is also this. Understood? So all the cells, as the cells of the tissue are very active, they're actively growing, they'll have dense cytoplasm, thin cellulose wall and prominent nuclei. Will they have vacuoles? No, because they're rapidly dividing. काम ही नहीं कर रहे ना प्रोडक्शन का और स्टोरेज का, तो तब करेंगे जब प्रोडक्शन करेंगे, उससे पहले तो डिवाइड कर रहे हैं. चाइल्ड मैरिज यहीं चल रही है सारी. New cells produced by meristem are initially like those of meristem itself. They are rapidly growing, slowly changing. Permanent tissues are permanent. Now we are specialized in our function. They are differentiated. There can be two types, simple and complex. Simple are basic one type of cell. Example, parenchyma, you can write here. Example, parenchyma. Next, complex permanent tissue, more than one type of cell. Example, xylem and phloem. Both are conducting tissue. Conducting means transporting. When I say that iron is good conductor of electricity, means from one end to another end, it can transport it, conduct it. Same way, xylem and phloem are transporting. So they are conducting tissues, right? Can I say them as vascular tissue also? Because they have vessels, tubular structure. Just like our circulatory system, blood vessels have vascular structure, means vessel-like structure, which can help in carrying or conducting the fluid. They are also like that. So they can be called as vascular structure. They can be called as conducting tissues. Xylem, they are made up of tracheids, vessels, xylem parenchyma, xylem fibers. What are tracheids? Tracheids are these structures. Tubular structure, pits are there so that water can move inside and they can flow. Same way, vessels are there. Next is phloem. Phloem has 4 types of elements: sieve tubes, companion cells, phloem fibers and parenchyma. Sieve tubes are perforated, means holes will be there which will allow material to go inside and flow. Next, another important difference between xylem and phloem is xylem is unidirectional, means from root to tip of plant water will move. It will not go in reverse direction. In case of phloem, it can go in both directions. It is bidirectional. For example, if a plant is there, leaf doing photosynthesis, and making glucose, it wants to store this glucose into root because root has become storage organ. Example, radish, मूली, carrot, in सब में. So this food can come in this direction. If it has to be stored in some fruit, this food can go in this direction. So it is bidirectional in nature. So phloem is bidirectional, xylem is unidirectional because water movement has to take place in only one direction. Bottom two, done. Plant tissue is done. Next is PYQs. अच्छा हाँ, बॉम्बे ब्लड ग्रुप में जो हमने किया था ब्लड ग्रुप्स, उसमें बॉम्बे ब्लड ग्रुप आई गेस रह गया था. जो बेस है, मतलब all the blood groups has been derived from a basic material or which is antigen, which is called as H antigen, which I told at that time. H modifies to form A, B, and slight modification forms O, but it does not express. Now, if it remains as H only, if it remains as H only and does not modifies to form other, then that blood group is H blood group, which is very rare and is usually seen in individuals of Bombay and first time visible there and was named Bombay blood group because of that, and it is present there. The reason is because the marriages are happening in a closed group which has this blood group. So therefore it is expressing there, otherwise it cannot express if ABO other blood groups are there. Done. Only this much you should know, that it's rare, first time found in people of person of Bombay, and it is not expressing ABO blood group. That's it. Now let's come to PYQs. The first one. किसने बोला B? Online. First, in the context of hereditary disease, consider the following statements. First, passing on mitochondrial diseases or disorders from parent to child can be prevented by mitochondrial replacement therapy either before or after in vitro fertilization. Correct, incorrect? Correct, we have done this today only. Second, a child inherits mitochondrial diseases entirely from mother, not from father. True, both A and B. Next, consider the following: bacteria, fungi, virus. Which of the following can be cultured in artificial synthetic medium? Viruses नहीं हो सकते. 3 जाते ही. Answer is 1 and 2. In the context of recent advancements of human reproductive technology, pro-nuclear transfer is used for. Pro-nuclear is after the fertilization, whatsoever the nucleus is formed, that is transferred to some other zygote to prevent mitochondrial disorder. 2020 में क्वेश्चन पूछा, अगेन माइटोकॉन्ड्रियल डिसऑर्डर से 21 में पूछा, राइट? दिस क्वेश्चन इज आल्सो रिपीटेड 13 में. Next, which of the following statements are correct regarding the general differences between plant and animal cell? First, plant cells have cellulose cell walls while animal cells don't. True. Next, plant cells don't have plasma membrane unlike animal cells. All cells have plasma membrane. Mature plant cells has one large vacuole and animal cells have many small vacuoles. Correct? वैसे तो second गलत होते ही. Answer is 1 and 3B. Right? Next, which of the following statements is or are correct? Viruses can infect bacteria, fungi, plants. All. It can infect everyone. Answer is D, 1, 2 and 3. Online students answer by mentioning the question number also. सीधा लगे हुए हैं B, D, C. Next, which of the following statement is or are correct? Viruses lack enzyme necessary for generation of energy. True. Viruses can be cultured in any synthetic medium. यही पूछा था दोबारा 2021 में, right? Viruses are transmitted from one organism to another by biological vectors only. हवा से भी हो सकता, only कहीं नहीं है. हवा पानी कहीं से भी हो सकते हैं. जब ऐसा कोई डाउट आए ना ओनली में, ऑलवेज थिंक ऑफ एन एग्जाम्पल. फॉर एग्जाम्पल, अगर कोविड वायरस है, हवा से हो रहा था ना, HIV कैसे भी हो रहा है, ब्लड से, ट्रांसफ्यूजन से या फिर किससे इंटरेक्शन से. सो यूज एग्जाम्पल्स ताकि कॉन्फिडेंस आए. यूल गेट कॉन्फिडेंस कि नहीं ये स्टेटमेंट गलत है, एक्सट्रीम स्टेटमेंट है. राइट. सो आंसर इज A, one only. Next, which one of the following processes in the bodies of living organism is a digestive process? See, this was given, the question was given for digestion, but what we have studied today, you can answer this question. For cell also, because the processes remain same across. Whatever is happening in the cell is happening in your body also. Conversion of glucose into glycogen. Digestive पूछा है. अच्छा, digestion के लिए which cell organelle we have studied? Which breaks down the components? Which cell organelle breaks down? Enzymes कहाँ है? Lysosome. Go to lysosome in your notes. Breakdown of proteins into amino acids. Breakdown देखो ना, पहले तो तुम ये देखो conversion कहाँ है? Breakdown का मतलब क्या? ऊपर क्या लिखा है? Digestion. Digestion means breakdown itself. So breakdown has to happen, right? Digestion में. Simple से complex बनेगा, complex से simple. Complex से simple in digestion. Now write here additional points besides this answer. Glucose is stored in plants in the form of starch. Glucose is stored in plants in the form of starch and in animals in the form of glycogen. And in animals in the form of glycogen. Done. So जब भी हमारी बॉडी में whensoever the amount of glucose is increased, so plants store it by the method of by converting it into starch. That's why you get starch from potato, rice. Why? Because they are storing it in the form of starch. Animal bodies store it in the form of glycogen. So whensoever extra glucose will be there, it will be stored in the form of glycogen, and more is there, then it will be stored in the form of fatty acids also. Convert हो जाता है और ज्यादा, और फैट्स की फॉर्म में भी स्टोर हो सकता है. राइट. नेक्स्ट, the cellular and molecular control of programmed cell death is apoptosis. We have done aging, degeneration, necrosis. Degeneration, there is no cell death, but the cells will start degenerating, means they'll reduce in size, they'll not perform much activities, but death has not happened. Okay. Next is, which of the cell organelle play most significant role in protein synthesis? B, endoplasmic reticulum and ribosome. Which organelle in the cell other than nucleus contains DNA? Mitochondria. क्यों? कभी बैक्टीरिया था. Next, which of the following statement regarding starch and cellulose are not correct? Both of them are of plant origin. Correct, because you know cellulose forms the plant cell wall. Cellulose forms the plant cell wall, and starch is found in plants. Glucose is stored in that form. So therefore, they are plant origin. Correct. Both of them are polymer. Yes. Both of them give color with iodine. Let's say you don't know it. Both of them are made up of glucose molecule. Yes. Both starch and cellulose are made up of glucose molecule. The arrangement is different. Okay. It is carbohydratic in nature. So the answer is C. Next, complete conversion of glucose in the presence of oxygen into carbon dioxide and water. Aerobic respiration. Alpha keratin is found in skin. Fat present below the skin surface act as a barrier. Loss of heat from the body. Next, match. Photosynthesis is done by chloroplast. One, mineral uptake, plasma membrane, respiration, and protein synthesis. 1, 2, 3, 4. Next, a person with AB blood group is sometimes called universal recipient because lack of antigen in his blood. ऐसा है क्या? A और B एंटीजन दोनों है, तो एंटीजन्स तो है ही है. Lack of antibodies in his blood. Yes. Yes, because बचपन से उसने A और B दोनों को देखा है. Doesn't have any antibody. So it will not react with anything else. It can accept blood from anyone. Presence of antibodies, अगर antibodies होती तो react करता ना. 13 का B, skin. 16 का B. हाँ, क्या कर रहे हो? Receive कर पा रहा है सबसे. So, when it is receiving from everyone means it is not reacting. If it is not reacting means it does not have antibodies, right? Hair में भी है, skin में भी है. Done? Class over.